Good morning, everyone. Welcome to the first session of HydroMediT. I would like to welcome you to the beautiful island of Lesbos. I wish you a pleasant staying. Our first presentation is integrated multi-trophic aquaculture with sea bass, sea bream, and mussels in the Aegean Sea by Kurtai and Locke.
Good morning, everyone. I'm Evrim Kurtay from Ege University Fisheries Faculty, Department of Aquaculture from Turkey. And today, my first presentation is about our study of integrated multi-trophic aquaculture with sea bass, sea bream, and mussels in the Aegean Sea. Let's have a quick look to philosophy of integrated multi-trophic aquaculture. Okay. Should sit down and go on like this. And the IMTA, integrated multi-trophic aquaculture, it can be simply defined as culturing more than one species from different trophic levels at the same time at the same aquaculture environment. Okay, then please click here. Again, the integrated multi-trophic aquaculture philosophy can be defined as culturing more than one species from different trophic levels at the same time at the same aquaculture environment, which can ensure both reducing environmental impacts of aquaculture and diversify products and also raise income with sustainable aquaculture practices. An IMTA method is very flexible and can be applied both inland and marine environments. Species from different trophic levels are being used like fish and shrimp, fish and mussels, fish and kelp, fish and sea cucumber, mussels and sea cucumber, or both fish, mussels, and kelp. The IMTA has advantages and disadvantages, but it has many advantages like providing production diversity by means of culturing more than one species at the same environment, and it reduces the possible risk of destruction of one population because of disease. It ensures more income for farmers when per culture area considered. It ensures efficient management applications in terms of waste reduction, oxygen intensity, environmental wellness, and animal welfare. It is more acceptable by public opinion regarding environmental friendly approach, and it reduces environmental impacts of culture operations. However, it has some difficulties and challenges, like it requires more knowledge in terms of culture methods and application practices of aquatic organisms, and it's hard to calculate carrying capacity of your culture environment or deciding the appropriate species to integrate. The results of our study that I'm about to present was recently published in the journal called Regional Studies in Marine Science, and this study was funded by Ege University Scientific Research Projects Coordination Unit with a number 1062. Our study site, as you can see on the slide, is not far away from the conference island, the Mytilene, and it can be referred as the midsection of the Aegean Sea. And the study conducted at a fish farm called Iskandil Fish Production, Food, Tourism, Trade and Industry Limited Company. The company produces both sea bass and sea bream, and the fish farm has 15 cages with a diameter of 20 meters each. And the total annual production of the fish farm is 210 tons per year. And to integrate the mussels, first we took water samples around the sea cages and to define the chlorophyll A concentrations, which is feed of mussels around and inside the sea cage system. But there was a question in our mind. Where is the impact point of fish farm in terms of effluent of waste feed, organic and inorganic matters accumulation, and how deep it occurs, and of course, how it affects the chlorophyll A concentrations at this spot. For this purpose, we took water samples at every 5th and 100th meters from the sea cage center, and every distance we took water samples from different depths as surface 10 meters, 20 meters, and 30 meters, which is bottom. You can see the chlorophyll concentrations at 100 meters away from the sea cage center according to depths. There was no statistically significance at 50-meter samples, but the water samples that was taken from 10 meters deep from south direction was statistically significant from other directions and depths. After that result, it was decided to locate one of the culture systems at 100 meters south and at a depth of 10 meters. According to the results of the chlorophyll A concentrations, 3 stations were decided to deploy experimental mussel culture systems. First, the center of the sea cage system were determined as station 1. The second station were determined at the south 100 meters away from the sea cage farm where the chlorophyll A concentrations was the highest. And the third station was determined at the 500 meters away from the sea cage farm at the east side, which was the chlorophyll A concentrations was found at the lowest. And to integrate the mussels, an anchor, rope, shackles, U-bolts, and a buoy were used to build the experimental mussel culture system. Cylindrical spot culture bags were built by a polyethylene mesh material with 5mm mesh size. The mussels were collected from the buoys and ropes of the sea cage farm and classified at the facilities of Ege University, Faculty of Fisheries, Department of Aquaculture in Urla, Izmir, very close to the sea cage system. Mussel spot with an initial length of 10mm were put in the mesh bags that have been prepared previously, and the mussel bags were created. Then the mussel bags were hung on the system that have been deployed previously at the stations. The shell length, width, thickness, and weight of mussels were observed for a year period. In addition, meat yield and condition index of mussels were also determined. And seawater temperature ranged between 14 and 26 degrees Celsius, and the annual mean water temperature was approximately 19.5 Celsius. The chlorophyll A concentrations were approximately similar between stations, but the annual chlorophyll A concentration was the highest at station 2. Likely with the chlorophyll A concentrations, the particulate organic matter, particulate inorganic matter, and total particulate matter was similar at all stations. According to one-way analysis of variance of length values, the second station was statistically significant from both stations in the following months, including November. There is no statistical difference between the first station and the third station. The mussel juveniles with an initial shell length of 10mm reached the highest at station 2 at the end of the study, which was a year time. And however, any statistical significance was not found. The mussel shell width values were higher than the other 2 stations at site 2. And similarly, the shell thickness of mussels were higher at citation 2, but again, any statistical significance could not be found. Although the increase in weight was similar at all stations, until December, a significant difference in the second station was found since January compared to other stations. The meat yield was higher than the other stations at station 2, but any statistical difference cannot be found in terms of meat yield. And the condition index is again the same, but there's a different, since August, the values were statistically significant until the end of the study in site 2. Here you can see the growth in shell length and weight gain of mussels in similar studies. We achieved more efficient growth values than other studies in terms of mussel growth at an IMTA study, but I have to point out that our study is the one study that has searched for the certain impact point of the sea cage farm. Researchers deployed their juvenile mussels to the vicinity of the sea cage farm or the center of the sea cage farm in previous studies. According to our studies, the impact of sea cage farming does not occur at a certain place, like the nearest position. Each farm has its own unique oceanographic and biological characteristics that should be evaluated in itself. Therefore, the flow rate, many parameters such as nutrients and oxygen levels, temperature and salinity changes, etc., must be known and can be monitored in the region where the integrated culture will be made. Thanks for your interest.
Okay, thank you very much. Are there any questions? Any questions? No. Okay. We can now proceed to the next presentation. The next presentation is the predictive value of high sensitivity C-reactive protein and ceruloplasmine for lactococcosis in rainbow trout by Mr. Zabrainova.
Seçer. Örneğin. Hello everyone, I am Sertal Seçer from Ankara University, Department of Aquaculture. Today I will present you our study titled as the predictive value of high sensitive C-reactive protein and ceruloplasmin for lactococcosis in rainbow trout. As you know, that bacterial diseases constitute one of the most important problems in the cultured freshwater and marine species in the world. It has been reported that the bacterial infections leading to mass mortalities cause economical losses in trout production. Pathogenic bacteria such as Lactococcus garvia, Vibrio angularium, Yersinia ruckeri, Aeromonas, Pseudomonas, and Phylobacterium have been frequently reported to cause disease and mortalities in rainbow trout. Lactococcus garvia causes infection named lactococcosis, which affects both freshwater and saltwater fishes and can be pathogenic to mammals, including humans, and typically distinguished by hyperacute and hyperemic septicemia. Most susceptible species in the rainbow trout, which is characterized by an acute reaction and increased mortality compared to other fish species. The secretion and function of acute phase proteins vary by animal species. the plasma concentration of some proteins increases, and these proteins are called positive acute fast proteins, such as ceruloplasmin and C-reactive proteins, while the plasma concentration of other decreases, and these proteins are called negative acute fast proteins, like albumin. In fish, the liver is the prime source of acute fast proteins. C-reactive proteins has been described in many species, inclusive of rainbow trout, and considered to be one of the most important acute fast proteins in the vertebrates, including the teleost fishes. Ceruloplasmin, as a peroxidase, has a major role in the protecting host tissue from toxic oxygen metabolites releasing from phagocytic cells during inflammatory states. In this present study, we aim to establish the alteration occurring in blood circulating acute fast proteins, high sensitive C-reactive protein, and ceruloplasmin, and to locate whether these two acute fast proteins can be used as early diagnostic markers for lactococcosis in rainbow trout. In our study, we take the samples from the fish farm in Fethiye in Turkey, which has a mortality rate varied from 15%. The bacteriological samples of kidney, liver, spleen, and the blood was straight onto the typical soy agar and brain heart infusion agar and incubated at 20 centigrade Celsius for 24 to 48 hours. After incubation, we isolate 15 of the 15 bacteria and bacteria colonies, and we recovered them and make some tests on them. The first one is the conversion method, as we know that the gram strain, the oxidase test, and the other test, the biochemical test actually. Then we make the 16S RNA sequence analysis, and also we use Maltov system to identify the bacteria that we isolate. As a control group, we took 15 fish and take the blood from them and get the plasma from their blood, and ceruloplasmin and the C-reactive proteins can be determined ceruloplasmin and C-reactive proteins. For the ceruloplasmin, we use the removing methods, which described by Beshuva and Kolb. And for the C-reactive proteins, we use fish high-sensitive ELISA kit, which was pre-coated with an antibody specific to CRP. After the incubation, there were 15 non-motile gram-positive coccus bacteria form chains, facultative fermentative, secrum oxidase, and catalase negative, and alpha-hemotic on the blood agar. We also recovered from the Maltov the score of the biotyper is between 2.4 to 2.1, which identicate highly in this device. And also according to 16S RNA profile, we identified our bacteria as Lactococcus garvia. And after that, we checked the ceruloplasmin and the C-reactive proteins on the plasma. And according to these data, we get the scores that we shown in the table. The control group is the diseased group's plasma's ceruloplasmin level is more than 6 times higher, and the C-reactive protein is a bit higher if we compare with the control group. The innate immune system of the fish participate in the response of the inflammatory provocators by activation of phagocytose and stimulating white blood cell activity and altering the values of the acute fast proteins. In the present study, changes in the blood proteins, high sensitive C-reactive protein and ceruloplasmin were observed in rainbow trout in response to natural infection caused by Lactococcus garvia. Acute fast proteins are synthesized during the acute fast response, which has a key role in restoring the homeostasis of the body. An acute fast response can be caused by infections, inflammations, variation stressors, trauma, or tissue damage. Generally, levels of C-reactive proteins in fish are relatively higher than in humans and mammals. The investigation indicates the presence of noticeable difference in blood results in rainbow trout with natural lactococcosis compared to the healthy ones. In the present study, the results revealed that the concentration of the C-reactive proteins in control group was lower than in infected fish. Also, blood C-reactive proteins values are slightly high affected by bacteria, but natural lactococcal infection has led to significant increase in the seroplasmin values. In the present work, it was observed that the natural infection of Lactococcus garvia have induced marked increases in seroplasmin concentrations compared to control values. The use of seroplasmin for diagnosis remains less frequent than that of C-reactive protein, but this suggests that this protein can also be used as an indicator of infection in this fish species. According to some authors, changes in the concentration of seroplasmin are related to the degree of infection, and the increase in the indicator is in response to the degree to which the liver is affected by pathogen, which is a reason for the synthesis of positive acute proteins. Seroplasmin may have some role in the body's antiparasitic defense, but furthermore and more detailed studies are required. Thank you for your participation.
Thank you very much. Any questions? No? Now we can proceed to the next presentation.
Okay, so now we call at the stand Mr. Curtain for the presentation of reproductive cycle of mussels integrated to a fish farm, IMTA, in the Aegean Sea.
Hello, everyone. It's me again. And second presentation is about the reproductive cycle of mussels, which is integrated to a fish farm. This maybe the second or third slides are the same, but the study is conducted at the same place at the same time. But this time, we search for the gonad development of the mussels. The study site is the same with the previous presentation. It's in the mid-Aegean Sea. And the water, while we are deciding the stations, they are the same, the water temperature, water chlorophyll A concentrations, etc. And these are the stations, as you know. And the environmental parameters are also the same. And now I want to talk about the method. The gonad samples were, when the mussels collected from the system and delivered to the laboratory, the process immediately started. And firstly, the epibionts of mussels were cleaned to prevent any corruption in gonad tissues. And after cleaning the mussels from the debris and epibionts, the shells were opened and gonad tissue dissected by a scalpel and immersed in Davidson solution for fixation for 72 hours. Gonad tissues were embedded in paraffin, and then the gonad tissues cross-sectioned to a 6-micron thickness with a Shandon Finis microtome, and the samples were immersed in xylen and alcohol series, respectively, then dyed with hemotoxylin and eosine. And the temperature, the mean temperature was about 19.5 degrees, and the chlorophyll A concentration was highest at the station 2. I'm sorry. And histological observations were performed by using an Olympus BX 51 brand microscopy with built-in digital camera system. The gonad development stages were identified in 4 phases according to seed, which are 1 resting, 2 developing, 3 mature, 4 spawning. And at the first stage, resting or spent, any phase of germ cells could not be observed at this stage, and gonad is consisted of only connective tissues. Sorry. And the second phase was the development stage. The first stages of spermatogenesis and oogenesis occurs in the development phase. Follicles are become visible in the connective tissue in both males and females. Spermatogonia and spermatocyte cells in males and previtellogenic and vitellogenic oocytes are become visible in females at this stage. Mature stage, follicles are full of mature germ cells and reach their biggest volume. In mature stage, the gap between follicles decrease. Spermatid and spermatozoa cells in males and vitellogenic and mature oocytes are become visible in females at this stage. And spawning, the last stage, which is spawning, gaps occurs in follicles and while connective tissue expanding between follicles. Number of spermatozoa and mature oocytes become decreasing in the follicles. The channel-like structures can be observed by dissolving and unification of the follicle walls. At site 1, the first developing muscles were seen in September. Mature and spawning individuals were seen till March. The big number of individuals was in spawning phase in December and January. At site 2, which is deployed in the impact point of CKH farm, both developing, mature, and spawning individuals were seen in October, but unlike site 1, the number of spawning individuals remained high throughout the 3 months covering December, January, and February. It may be thought that having more nutrients at station 2 compared to station 1 encourages more individuals to spawn. At station 3, the big number of mussels started spawning in December, which is late from other 2 stations. But nearly all individuals in the population was in spawning phase during January and February. Gonad index were calculated according to method of Soria. According to this formula, the gonad development stages were categorized in 3 by coefficients as 1 for resting or spent, 2 for early, active, and developing, 3 for mature and spawning. Gonad index was calculated by multiplying the number of muscles belonging to each category by the category coefficient and summing the values, then dividing the outcome value by the number of total individuals as follows. As it's seen on the graphic, the gonad indices were low throughout the summer season due to high water temperature. By the end of the September, water temperature started to decrease, and from October, gonad indices were started to increase, peaked in winter, especially December, January, and February at all stations. However, station 3 is peaked later than other stations. Any statistical differences could not be found between stations. A negative correlation was detected between temperature and gonad index values at all 3 stations, which is similar with previous studies of mussel gonad development. And gonad index increases when temperature decreases below 20 centigrade in the current study. The mussel spawning occurs in winter time, generally when the water temperature low. The studies in Spain were carried out in regions located in the north compared to our country. It's known that gametogenesis of mussels is faster at low temperatures and slows down and stops at high temperatures. In gonad studies conducted in Spain, the water temperature was 10 to 15 and was similar to that measured during the spawning period in our study. As the water temperature increases, there is a decrease in the spawning activity. It's known that decreasing in the temperature below 20 centigrade degrees starts the gametogenesis. After gametogenesis begins, the increase in temperature accelerates. The scission process in the development phase. In the current study, female-male ratios were found close to each other. It's considered that the number of male individuals at station 3 is higher than the female individuals because the progeny activity at the third station continued in March, when the study was terminated also, and the number of male individuals was higher. In addition, it has been reported that the population of mussels starved in laboratory conditions tend to be male, and the metabolic needs of male individuals are higher than the male individuals. Female individuals are higher than the male individuals. Since the station 3 has the lowest nutritional values in terms of chlorophyll A and TPM values, the fact that male individuals are more numerous than female individuals supports this situation detected in previous studies. Thanks for your interest.
Thank you very much for the presentation. Are there any questions from the audience? Okay, thank you very much.
You're welcome.
So now I'm going to call to come to the stand Dr. Sofia Papou, and she's going to present us her work with us also, the effects of algal dietary supplementation on the mussel biochemical composition and fatty acids of Dichedrachus labrax fingerlings.
Όχι. Ήρθε στο... Good morning and welcome from me as well. Thank you. Good morning, welcome. So my name is Sofia Papou. I'm a postdoctoral researcher in the Aegean University in the Department of Marine Sciences. So the work that I'm going to present to you today, which is not moving, the page is not moving. It's not moving. Next. So the objective of our study was to examine the effect of different doses of an antioxidant extract of vulva lactuca as a dietary supplement on the flesh biochemical composition of European sea bass. The need for such a project was built on the fact that during the last decades, the aquaculture production has reached a record high, and also it has a critical contribution at the food security and nutrition of the 21st century. Of course, two of the main factors for a successful industry in aquaculture is the nutritionally balanced diet of the fish and also the low production cost. Towards that, companies have turned into the use of functional feed additives. Functional feed additives can be a various of substances such as prebiotics, probiotics, seaweeds, microalgae, and other phytobiotic compounds, not only to improve the fish growth, but also the fish health. In particular, macroalgae have been widely used over the last decades as a feed supplementation in feed, either to substitute the costly fish meal and fish oil, or as therapeutic agents or growth promoting factors. Okay. So it has been stated in previous research that even though macroalgae are being widely used, there are certain plant-based products that must be processed before they can be used in aquaculture fish additives. For that reason, specifically, let's say European bass, which is a carnivorous fish, and other carnivorous fish species, they have a problem digesting raw algae material. So it has also been stated that through the selection of the extraction method that is used to disrupt the cell walls of the algae, the biological properties of the seaweed extracts can further be species-specifically adjusted. In particular, species of ulva biomass have been, as I said before, investigated over the last decades. But in the form of an extract, the studies are more scarce. And especially because the studies that talk about the use of ulva extracts are based on their polysaccharides. Ulva does not contain only polysaccharides, it contains a lot of other bioactive compounds, and especially also phenolic compounds and carotenoids and other pigments. What we did was to have an optimization extraction process on ulva lactuca. Our results have already been published, as you see on your screen. So we took this high antioxidant activity extract of ulva and used it on the fish feed, the commercial fish feed that we use for the European sea bass. We moved on to create 3 batches of fish feed. One was the control fish feed, the commercial one. And then we had 2 more batches, one with 1% addition of the extract, and the second with 3% addition of the extract in the commercial fish feed. About the fish and rearing conditions now, we had 450 juvenile fish with a main weight of 7.6 grams each, allocated to 9 tanks, 9 plastic tanks of 1,000 liters each. We daily fed them for 56 days. This is about 2 months. And the feeding was 6 days per week and at a ratio of 1.5% of their body weight. The water temperature, salinity, pH, and dissolved oxygen, of course, was monitored daily. And the sampling procedures took place in day 0, day 28, day 56. So at the beginning, at the half of the experiment, and at the end of the experiment. Prior to the sampling, the fish were fasted for 24 hours. And the sampling took place with using light anesthesia with phenoxyethanol. We proceeded to conduct a muscle proximate analysis on the edible part of the fish, which means moisture content, mineral ash content, total fat content, and total protein content. And then we moved on forward to use gas chromatography in order to identify and quantify the fatty acids of the flesh. So our results. First of all, in order to evaluate the effects of the antioxidant extract of ulva on the fish, we compared the control population to the 2 supplemented diets on day 28 and 56. All diets were equally accepted by the fish. There was no significant increase in the mortality rates in any of the tanks. And also, there was no significant differences between the fish morphological parameters observed. But we have to note that the population that was fed with a 3% of addition of ulva extract, we observed at the end of the experiment a higher percentage of weight gain and higher percentage of specific growth rate. The population that was fed with a 1% feed had the lowest. About our fish protein content, both supplemented diets exhibited a decrease in the proteins. And more specifically, the control population had an increase of about 4% in the fish total proteins, while the other 2 supplemented diets had the result of a decrease of about 33 to 34% in proteins. The mineral ash content was found to be higher for the 2 supplemented diets, although the control population had a decrease of about 23% in the mineral ash content of the muscle tissue. The fish that were fed the 2 supplemented diets kept their levels of ash stable. Finally, the total fat content was higher in fish that were fed the 3% inclusion during the first month of the trial only. And at the second month of the trial, we had a 10% decrease in the total fat content, while the population that was fed the 1% inclusion of ulva extract exhibited a 29 increase during the second month only of the experiment. Regarding the fatty acids, we were able to successfully identify 90 to 95% of our peaks in our chromatograms. We noticed that oleic acid was detected only for the fish that were fed the 2 ulva lactuca supplemented diets. And also we noticed that the population that was fed the 3% inclusion of ulva had significantly higher amounts of palmitic acid, eicosapentaenoic acid, and DHA. Overall, the control population flesh consisted of mostly 20% of palmitic acid and 60% of stearic acid. The 1% population consisted of 20 to 40% of palmitic, stearic, and oleic acid, while the 3% population consisted mostly of almost 50 to 51% of palmitic acid and almost 24% of DHA. In conclusion, we noticed that the 3% population that had the ulva dietary inclusion resulted in the highest observed specific growth rates and weight gain, the higher, almost 50% higher amounts of mineral ash content in their flesh, and also they had higher concentrations of myristic, palmitic, eicosapentaenoic acid, and DHA. Oleic acid, again, was detected only for the 2 populations that were supplemented. And what we have to suggest for a future similar study is that a higher daily feeding rate or a more prolonged experimental time would be suited. And also the addition of the analysis of hematological parameters would provide us information in order to have a more clear insight on the mechanisms that worked behind the observations that we've got. Thank you very much for your attention.
Thank you very much, Sofia. And I would like to ask the audience if there are any questions. Yes, one question here.
I would like to congratulate the team. That's a very great job you did. And it's somehow close to our background regarding the extract. A curiosity, why do you think the total protein content decreased in the fishes fed with ulva?
Excuse me, can you repeat the question?
Yes, why do you think the total protein content decreased in the fishes fed with ulva?
With the analysis that we conducted, we do not have enough data to come to a sure conclusion about that. That's why I suggested that we would need more data and more analysis on that, which is actually what we are about to do. We keep working on this.
Great, thank you.
Thank you very much.
Okay, any other question? Okay, thank you very, very much, Sofia. And I would like to welcome to the stand Dr. Locke, and she is going to talk about the reproductive cycles of Olothuria tubulosa and Olothuria poli in the Aegean Sea coast of Turkey.
Thank you, Chairman and contributors. I would like to share our results on reproductive cycle of sea cucumber in Aegean Sea. Sorry, it doesn't work. Yeah, there's a problem here. Mouse, yeah, the problem. Ah, with the mouse on. Okay. Down. Okay. For the introduction, the sedentary nature of sea cucumbers, the high volume and demand in the international market, and the low cost of processing make sea cucumbers very vulnerable to overexploitation. Sea cucumbers have used as food source and medicinal substances for centuries, mainly in Asian and Middle Eastern countries. Overfishing in recent years has led to local extinction of high-value species in some localities and prompt closures of many national fisheries to allow stocks to recover and to allow more sustainable management plans to be established. Holitoria tubulosa and Holitoria poli are the most targeted species in Turkey. The population density of these species in Turkey's Aegean coast has decreased tens of times in the last 5 years. Studies on reproductive biology of these species are important for sustainable stock management. The main goal of this study is to determine reproductive cycle of tubulosa and poly in Aegean sea coast of Turkey. For the purposes, specimens of 2 sea cucumbers were collected monthly from Urla, İzmir Bay between November 2018 and October 2019. In each sampling period, randomly 30 individuals for 2 species were collected by divers and brought to the laboratory and with plastic containers and filled with seawater. At the same time of sampling, seawater temperature and salinity were recorded. In the laboratory, they transferred to the cuvettes containing seawater when their contractions stopped and relaxed in the water. The length and total weight of each individual were measured. Then all specimens were dissected and their gutted weight and gutted length were recorded after the removal of viscera. Gonad samples yes. Gonad samples were used to calculate the gonadosomatic index to determine sex and gonadal development stages. Gonadosomatic index was calculated using gonad weight and gutted weight. For the reproductive cycle, the gametogenic stages were assessed by using histological examination. Gonad samples were fixed at 10% formaldehyde and embedded in paraffin. 5 to 7 micrometer sections were cut on a microtome and stayed with hematoxylin and eosin. Gonadal development stages were determined and photographed under microscope. Gonadal stages has been examined in 4 stages: development, ripe, spawning, and spent. During the study, the average water temperature was 18.5 centigrade, and the highest temperature was recorded in July and August. The salinity volume did not change significantly, and the average was 35.5 ppt. Since sea cucumbers have worm-like contractions and relaxation movement and retain a certain amount of water within the body, length and weight measurements may not be stable. In this study, weight and length measurements were taken both while they were alive and after their internal organs were removed. The result of the measurements for both species are given on the table. Due to random collection, their sizes varied by months. Sex ratio of tubulosa, Holtoria tubulosa, differs significantly from 1 to 1 ratio, demonstrating a ratio of 0.65 to 1.50, with a higher proportion of males than females in the most months. However, the sex ratio of Holtoria poli is the opposite of tubulosa, and the number of female individuals was always significantly higher than the males. Gonadosomatic index values are indicators of gonad maturity and reached the maximum value in July with 20% in tubulosa, while the average gonadosomatic index reached its peak in May with 28% in poly. In both species, it decreased to minimum values in the autumn and winter months. Although Holtoria poli reached its maximum gonad productivity in May at a temperature of approximately 20 degrees. Holtoria tubulosa picked its gonadosomatic value at a higher temperature, 25 degrees. As seen in the figures, while gonad development was observed in late autumn and winter months in both species, the maturation rate of gonads increased in the late spring. The spawning period for both species coincided with higher temperatures in summer and continued till October. If we compare our results with other researchers' findings, the reproductive period of Holtoria tubulosa is similar to the reports of researchers from northern Aegean coast of Turkey, Adriatic coast of Croatia, and western Aegean coast of Greece. However, in Algeria, spawning occurs at lower temperatures such as 19 to 20 degrees. The sex ratio in this study differs significantly from the other findings. While the female ratio decreased in tubulosa population, the male ratio decreased in the population of Holtoria poli. In conclusion, year-round water temperature variation is favorable for the gonad development of sea cucumber in Izmir, while the Holtoria poli matures in its gonads and starts spawning in late spring when the water temperature reaches 20 degrees. Holtoria tubulosa prefers the summer months when the water temperature is higher. Exploitation of the population for commercial purposes by overfishing causes the sex distribution to become unbalanced. Unless the fishing pressure is reduced, it is obvious that the continuity of the sea cucumber population in Izmir Bay is under risk. Thank you for your listening.
Thank you very much for the presentation. Are there any questions? Yes. The lady over there, and then the...
Hello, my name is Ioanna Argyru. I would like to ask you if you have, according to your legislation, any ban for the fishery of Holothuria and to what periods? Thank you.
Yes, thank you for your question. Yes, we have a ban and between 15 April to end of October, 31 October.
Yeah. Thank you, Dr. Locht, for your nice talk. I would like to ask first, because you said that the population of these species are in a risk. And if you tried in your laboratory to control the reproduction and produce Holothuria in your lab?
Yes, thank you again for your question. And we have some of research in our university to support our population. So one of colleague succeed to produce from egg to a commercial size of sea cucumber in our laboratory in Aegean Ege University. Which? Yeah. Yes, yes, we have aquaculture facilities in university, and we collect mature ones from the wild stocks. We spawn in our controlled conditions. Yes. Okay. Is there any other question?
Thank you very much again. And I would like to ask Dr. Berylis to come to the stand in order to present us his teamwork, effects of dietary supplementation with various mushroom species and yeast products on the liver and intestinal histomorphology of gilded sea bream. Your stand is yours.
Okay, thank you very much. Hello, everybody. I would like to present our work, effects of dietary supplementation with various mushroom species and yeast production on the liver and intestinal histomorphology of gill-heeled sea bream. First of all, liver and intestine are very important organs for the fish, for nutrient metabolism and absorption, and for the metabolism and detoxification of toxic substances that may be contained in the feed. So histological examination of this organ is important in order to evaluate the effectiveness of fish diet, and by describing tissue changes, we can detect any possible pathological changes caused by dietary modification. When dietary lipid is excessive or less digestible, large synthesis and deposition of triglycerides in vacuoles can take place. So distinct lipid droplets in the hepatocytes can cause displacement of the nucleus to the edge of the cell, and the excessive accumulation of intracellular fat can cause hepatic steatosis. The anterior gut is the primary site for the nutrient uptake. The microvilli, the brass border of the apical surface of the enterocytes, can increase the area of absorption, and dietary nutrient manipulation can cause numerous lipid droplets in enterocytes. They can be located mainly in the supranuclear position and can cause also some inflammations like enteritis. Mushroom and yeast are rich in polysaccharides, monopolisaccharides, B-glucans, and chitin. They have been proved to be functional nutrition supplements in fish diets with beneficial effects on growth and immune response. The aim of our study was to evaluate the effectiveness on liver and intestine structure via histopathology. So, therefore, we made 9 isonitrogenous and isoenergetic diets. As you can see to the table, the first one was the control diet. It didn't contain mushroom or yeast supplementation. Then we have 3 diets contained mushroom supplementation, and the other 5 diets contained yeast supplementation. The AG diet contained 2% of dried powder of Agaricus dispersus. The PO diet contained Okay, the PO diet contained 2% of dried part of portobello. PL diet contained 2% of dried powder of pleurotus. GTY diet contained 2% of grain distillers dry yeast. BY diet contained 2% of brewery yeast. AY diet 2% of autolyzed yeast. MOS diet 0.05% of commercial premium yeast fraction from the Saccharomyces cerevisiae, and the LY diet 0.25% of a commercial live yeast product. We use Parvus aurata juveniles, 3.3 grams initial mean weight. We use triplicate groups, 22 fish per tank, and of course 3 tanks per dietary group. Fish were fed to satiety twice a day, 75 days, each of the 9 diet. And at the end of the trial, liver and anterior intestine samples of 5 fish per tank were taken for histopathological examination. We use section of 5 to 10 micrometers, and we examined them under a microscope equipped with a digital camera. We used a semi-quantitative scoring system in order to quantify the histopathological changes. Grade 0 was no remarkable changes. Grade 1 was minimal changes. Grade 2, mild changes. Grade 3, moderate changes. And grade 4, severe changes. We also measured, by the use of IMAGE, the height of the anterior gut's brass border, the goblet cells diameter, and the number of goblet cells per 100 micrometers of intestinal fold. The supplementation of yeast or mushroom did not improve fish growth. Fish had similar final weight with the ones fed the controlled diet. Liver showed normal histological structure for all dietary groups. The grade that we recorded was 0 to 1. Hepatocytes nuclei appeared normal and central, and pancreatic isolates were prominent, and within the acinar cells, there were a large number of eosinophilic secretory zymogen granules. Only few and small lipid droplets were detected in the liver, and there was absence of hemorrhages. Here you can see 2 histological images of the AC diet and the AG diet. You can see that the liver structure is normal, and in the second picture here, you can see that there are few small lipid droplets. For the intestine now, all samples contain the normal intestinal structure. In the intestinal epithelium were detected enterocytes with distinct brass border. We detect also goblet cells, blood vessels, and scattered absorbed granules in the cytoplasm of the enterocytes. However, there were some significant differences in the intestinal structure. Again, 2 histological images of the anterior gut, the GTY diet and the PL diet. They were both had the normal structures of the anterior gut. And here are the differences that we observed. Fish fed with the mushroom or yeast supplemented diets had significantly higher brass border height than the control. In this table, with green color, you can see the lowest value, and with the purple color, you can see the highest value. We are here in row number 1. The control group showed a trend for larger goblet cells, but this was significant only when compared to AG, LY, and BY groups. Finally, LY group fish had significantly lower numbers of goblet cells compared to PL, GTY, BY, and MOS groups. The existence of few and small lipid droplets in the liver for all dietary groups indicates that there was an efficient lipid metabolism and a non-excessive intake of fat through the experimental diets. Fish fed with the mushroom or yeast supplemented diets showed a trend of higher brass border, and so there was a tendency to a greater absorptive surface of the enterocytes. These groups also showed a dread for more goblet cells in number, although the goblet cells were smaller in size, and so there was a tendency to strengthen the mucous intestinal epithelium that enhances the organ's protection. Similar to our findings, Espinosa and Esteban used dietary yeast supplementation, and they showed that sea breams tend to recover faster skin woods and reduce inflammation signs. Fronte also used autolyzed yeast and showed that it can lead to bigger absorptive surface of the anterior gut of the gill-head sea breams. And Dimitroglou used dietary mannan olysaccharides supplementation and showed that this supplementation enhances the absorption capacity of sea bream's anterior gut, and the brass border head was also increased. The digestive juices and nutrient absorption can be weakened by the excess of mucus in the anterior intestine, but the presence of mucous substances in the intestine can possibly regulate the transfer of proteins, ions, and fluids. Grau et al. Reported that neutral mucous compounds of the intestine participate in the enzymatic food digestion, formation of food mass, and the absorption. The number of goblet cells in the intestine can be dependent by the used diet. Martinez et al., when replacing fish meal with carrot throat meal in the sea bream diet, measured 5.8 to 7.5 goblet cells per 100 micrometer of intestinal fold, while Fronte et al. Measured higher goblet cell density in the anterior gut of the sea breams fed autolyzed yeast. Our results were similar and in agreement with Fronte, as we detect a tendency for higher goblet cell density in the anterior gut of fish-fed diets with yeast, except from those fed the LY diet, and we suggest that there is a link with the enzymatic food digestion and absorption. Thank you for your attention.
Thank you, Dr. Berylis, for your presentation. And I would like to ask again the audience if they have any question. Yes, the gentleman over here.
First of all, thank you for your presentation. I have a question. In your study, you studied fat cells in the liver, right? Right. Which type of microtomes did you cut samples?
Sorry, can you repeat? Can I hear you?
Which type of microtomes did you cut samples?
You asked me about the histology, right?
Rotary microtomes or freezing microtomes?
No, it was classical histological sections. No chromatomes. We used formaldehyde, and then we sectioned 5 to 10 micrometers and stained with hematoxylin eosins. It was classical histological examination, not cryomicrotome.
Don't use microtomes.
No chromatomes.
Oh. Okay, thank you.
Any other question? Okay. Thank you very much, Dr. Berylis, and thank you very much for your attention, ladies and gentlemen. The first session is finished, so we welcome you for a coffee break outside. Okay? Thank you very much. Αν κάθομαι. Ξεκινάμε το πρωί, δεν κάνω κάτι.
Hello. Welcome. Hello. Oh, very much. Oh, you arrived today. I mean, oh, wow. Presents. Yes, yes. Yes, yes. Thank you very much. Did you arrive today or yesterday? Yesterday. Ah, okay, okay. There are no direct flights here. It's a small island. Yeah, okay. Let's go outside. Να ρωτήσω κάτι. I will find you in a minute because I need to talk something about the... See you outside. Okay. Sophia is outside, Daniela, I suppose, Dan, everybody's out. Okay. Να ρωτήσω κάτι. Το να κάθεται το Προεδρείο εδώ πέρα, ας πούμε, και το κεφάλι του να αντικατοπτρίζει, να φαίνεται εδώ πέρα, μήπως μπορούμε να κάνουμε κάτι, ανέβει από το προβολέα, όχι, έτσι είναι fixed. Δεν μπορούμε να μειώσουμε λίγο, όχι, δεν μου λέει το πρωί, δεν κάθεσα εδώ πέρα και... Εντάξει, οκ, αυτό το γίνεται. Δηλαδή θα μπορούσε να μικρύνει το μέγεθος και να ανέβει λίγο αυτό, να μπορεί ο κόσμος να βλέπει εδώ, χωρίς να βλέπει τα κεφάλια μας από πίσω. Ναι, δεν γίνεται, λέει ο... Έχει όμως τα καλώδια. Έχει την εγκατάσταση από κάτω. Όχι, εδώ θα το πάρω. Δωράκια στο στόμα μου. Ναι, το πήξα. Ναι, να σε ρωτήσω.
Rick.
Good morning. Can I have quiet, please? Yeah. Good morning, and I'm welcoming as well. We have the section here, which is marine diversity and conservation. And I would like to ask the colleague and friend of mine, Professor Feliz Khalid, to come and give the first talk on from the predator to floating corpses, threatened Selachians in the Turkish Seas. Welcome.
Hi to everybody. Allow me to briefly introduce myself. I am Feliz from Faculty of Fisheries at Koçman University. My concern is fish and interested in project fish data providing to fisheries. I also interested in the habitat conservation and invasive fish species. I am here to talk about what's going on with shark species in Turkish waters. I think this study gives an overview of the threatened shark species in the Turkish seas. Also, it highlights the current status of the number of species that are protected by our national laws. Okay, thank you. Firstly, it's crucial to determine the number of shark species in Turkish seas in the light of current literature, completion and meetings with the subject experts state that 36 out of 48 sharks in the Mediterranean were also found in Turkish seas. Our extensive study to comprehend the state of shark conservation Turkish Sea, we evaluated 36 species using Mediterranean red list of the International Union for Conservation of Natures. To augment our research, we considered international agreements that the Republic of Turkey is part of. This consists of Barcelona Convention, Bern Convention, and Convention of the CITES. We incorporated also the national laws of Republic of Turkey in our study. Our primary goal was to compile a comprehensive list of regional shark species, particularly those classified as critically endangered, and vulnerable. Following our evaluation, we assigned a list of threatened sharks in the region. We addressed this by analyzing the number of cartilaginous fish caught in the region from 1967 to 2012, as reported by the Turkish Statistical Institute. Here is the deal. We've got 22 shark species in Turkish seas that are in trouble, making up more than 60% of the 36 species found there. As you can see in the table, 11 of them are pretty critical situation. Those are the ones highlighted in red. Also, we've got 6 species that are endangered, marked in orange. And finally, 5 species are just hanging in there, classified as vulnerable. They are ones that are highlighted in yellow. So when it comes to international agreements, only 6 out of 36 species we looked at were included in Bern. Now, if we consider just the 22 threatened sharks, Bern only covered 6 species. Regarding the international agreements, again, Barcelona had a bit more coverage with 20 species. When considering the 22 threatened sharks, Barcelona had a bunch more with 19 species. In view of international agreements, again, CITES was in the middle with 15 species. When considering the 22 threatened sharks, CITES covered only 9 species. When it comes national laws, the government, of course, play an important role in fisheries in Turkey. The government regulates fishing and other aquaculture production with the Fisheries Law 1300. It updates the regulations by creating notifications every 4 years. Notifications are mostly based on direct regulation. For example, they include issues such as minimum catchable size, fishing season, protected area close to fishing, species prohibited for fishing, etc., etc. These are the notifications, and since 2006, Turkey has been writing a basic national law to protect the shark species in our seas. Over time, particularly after National Action Plan meeting in 2017, the number of protected shark species increased from 5 to 12. As a result of our robust scientific efforts, 13 shark species have recently received national protection. Therefore, if we refer back to the initial point considering the values provided in slide 8, On the flip side, now national legislations covered 17 species, which is about more than 45%. Meanwhile, regarding 22 shark species, as we did before, national legislations included 15 species. As apex predators, sharks are integral to our ecosystems and play a crucial role. They regulate the species below them in the food chain and act as an indicator of a marine ecosystem's health through the removal of weak and sick individuals and balancing the competition. Sharks promote species diversity. This excellent infographic demonstrates why a robust shark population corresponds to healthy marine life. But sharks with 400 million years of experience are master of life. Scientifically, they are considered as slow growers, a point of which scientists agree, absent any natural predators excluded for human now. They grow slowly, reproduce at an older age, and produce few offspring. These traits, which make them the dominant force in the natural food chain, become their downfall when humans enter the picture. Consequently, basically, most sharks are pretty much at risk of all types of fisheries, whether they are coming at them straight up or a roundabout way. The IUCN reports reveal the concerning trend of over a quarter of the world's cartilaginous species are nearing extinction. This claim is supported by recent studies that show a significant decrease in the population of various chondrichthyan species in the Turkish seas. Once common and flourishing, these species are now, unfortunately, became increasingly rare. During the 19th and 80s, nearly 8 shark species were primarily targeted by Turkish fisheries. However, in today's Turkey, no fisheries currently operating along the Turkish coast directly target chondrichthyans. But cartilaginous fish often end up as bycatch in coastal and demersal fisheries. As seen in statistics, we statistically recorded only the Keller, angel shark, and top shark, and the graph indicates very decreasing the catch amount of the chondrichthyan fishes. And one of our studies in, I think, the 2007, we did this study where we wanted to see how much stuff get thrown away in a year from commercial bottom trawling. After a whole year of checking it out, we saw that trawling caught a whopping 85 different groups. Most of the catch, if you go by weight, was actually bycatch cartilaginous fish. The real bomber throat, more than half of stuff we had to throw back was cartilaginous fish. So, laws are super important when it comes to keeping threatened and endangered species safe. Take Turkey, for example. They signed on to some big-time agreements like the Barcelona, Ban, and CITES conventions. Right now, national laws in Turkey are doing a solid job for protecting nearly half of all shark species and over 2/3 of threatened sharks in the Turkish seas. The Barcelona Convention is doing the heavy lifting for shark conservation, protecting more than half of the shark species and whooping nearly more than 85% of the threatened ones. The Turkish science community and decision makers, top late, have taken the bull by the horns. 3 workshops and 3 committee meetings have already been conducted. I extend my gratitude to everyone for their non-ignorable efforts. The research carried out by a selected group of experts in Turkey territorial waters has resulted in publications that significantly contributed to ongoing regional red list assessment of cartilaginous fish in Turkey. The findings known by different names in the different regions and their inclusions as references in the species evaluations underscore our interdependence. The IUCN says we gotta check species status at least every 10 years. So in line with that, we can expect national assessments to beef up the protection provided by national laws. This means we'll likely see more threatened shark species getting the protection they need. And if we are gonna protect the shark species in our waters, here's that we gotta do. Map out their critical habitats, count how many are there out there, get to know their lifestyle, put together a comprehensive archive and dataset, make sure everyone involved on the same page, and get the locals in the know. Trust us, just depending on national laws, on international agreements, isn't gonna cut it. Thank you very much for your attention and interest. Yes.
And we continue with the next talk, given by Osgul, if I pronounce right, on strategic infrastructure for improved animal tracking in European seas, the Turkish Straits Array.
Okay. Hi everyone, I am Ichthyology from Ege University. It's okay? Hi again, I am Ichthyology from Ege University, opposite of shore. I will talk about the European funded project, the name of the straits today. Before, yes, mouse, but okay. Yes, but before the straits, a little talk about the acoustic telemetry. Acoustic telemetry is an aquatic tracking method that works to principle detecting signals from the transmitter, which place the aquatic animals by acoustic receivers, and is widely used to determine the behavioral patterns of the aquatic animals. We put some transmitter inside the fish or on side of the fish, and then detect the signals come from the transmitter with receivers or hydrophone. It's a new technology, but almost a very common method for tracking any aquatic animals. And also, this technology has developed last 50 years. A lot of acoustic receivers deploy around the world. You can see in the slide a lot of acoustic arrays, the Ocean Tracking Network, ETN, the Balearic Islands, and others. And in the Mediterranean, is a big hole. You don't find any receiver to tracking any animals. And this array collaboration with each other, and you can track the animals, the migration, high migration spaces, especially. So the ETN organization, ETN, European Tracking Network, was established 6 years ago. The aim of the ETN collaboration, European telemetry researchers together, and also tracking the aquatic animals in European seas. The ETN coverage, you can see in the picture the ETN coverage, and also you can see in the Mediterranean Sea and the Black Sea and Ocean, and activity of the ETN, results of the ETN activities, the CITRACE project was born. In the CITRACE project, we can put research infrastructures for important arrays. We name CITRACE. The Gibraltar, and the Irish, and Denmark citrates, and also Turkish citrates. The citrates strategic infrastructure improvement animal tracking in European seas, the name of the project we called citrates, is a horizon infra program. The focusing research infrastructure in the Europe is almost 4-year project. Is the budget is 3.5 million euro. We have 10 partners. 10 partners from 7 different countries. You can see the European countries and also ocean tracking from the Canada, and we responsibility Turkish trace as a Ege University. Our mission coordinate aquatic animal tracking and environmental observation efforts to scale that we usable to make a progress in international marine management and planning, expand the efforts to strategic connect innovative on species-based biodiversity management while developing data management plans and network channels as a step toward the permanent large-scale infrastructure for animal tracking in Europe. The objectives deploy active and passive acoustic infrastructure in 4 corners in Europe, undertake research projects that are developed and implemented collaboratively, strategically, and large scale, and Irish Danish citrates, North Channel, Gibraltar, and Turkish citrates. We focus to the 4 important traits. And also, we can, in addition to tracking important ecological and economical species, we can also tracking with the sea mammals with passive acoustic monitoring. In the Danish citrates, the DTO responsibility of the citrates, and they active now, and 48 receivers deploying the Danish citrates, and they also take Atlantic salmon, Baltic sturgeon, Pomrec rays, and also bluefin tuna. The simulator array is near the England and Ireland. Is a 140 receivers. They also tracking salmon, European eel, basking sharks, and other species. The Gibraltar citrate, 2 Spanish institutes lead this array. They also track Atlantic bluefin tuna, migray, Pomrecs, and blue sharks. Turkish citrates, the Ege University managed this array. We also deploy 25 receivers, the bluefish, Atlantic bluefin tuna, and European eel, and also some sharks and rays we will tag. Our impacts, for the first time ever, Europe's 4 corners will be instrumented to track underwater world. We can now track the animals sufficient scale to really access to understand their needs. This in turn can help to inform management and policy to better conserve and protect them. You can see in the pictures, the orange point is the citrates receivers, the blue one is the Etian arrays, and we almost covered the European seas. Not enough, but the starting point. And what is the impact? For example, you can show the picture in the bluefin tuna. We can understand the better estimate the bluefin tuna migration in the Mediterranean Sea. And similar is also the European eel. More than 1,000 eel have been tagging last 6 years, and you can understand the migration route with using this threat and Etian receivers. Now, I will give more information about the Turkish array. We deployed 25 receivers last year, and also temperature, oxygen will be measured continuously in Istanbul and Çanakkale citrates, and also we deploy passive acoustic monitoring devices on the Çanakkale citrates. And in the picture, you can see in the red points our receivers. It starts almost North Agency, continue Saros Bay, and also Çanakkale citrate, the 2 sides of the Marmara Sea, and also in the Black Sea, close to Bulgarian border and also Greece border. Yes, we started last year with some pictures. First, we have a meeting with the fishermen and ask which place is a good for putting some receivers and safety and also detection the species. We mainly focusing tracking the bluefish because it's important species for us. And then with some primary studies for sea bottom and the underwater situation. So this is a receivers and mooring system. This is a receiver underwater, and also Cassandra come from the ocean for support us. We can put a lot of place for receivers, fish farms, some mussel farms, some artificial reefs. And we will track the this kind of species, but first we start the bluefish because bluefish is a migration route. They come from the agency and return to Black Sea for reproduction. So we will plant 200 bluefish tagging. Now we almost 28 animals we take them, and also some picture from some video from. And surgery, we can some special table for surgery. The fish is anesthetic, and then cut, and after the insert the tagging, we another operation. You can see, and after
Elle bırakmak daha iyi zaten. Ben de göreceğim hocam.
It's a free. We are instrumenting the four corners of Europe. You can use this data. You don't give the money, only the member of the ETN, and you can use all the data. This is also good for the project because if you get a budget for only tagging the fish, you can use this infrastructure without any money. And also, if you need some training or education, the CITRICE team help you for how is the tag or how is the use data. This is also good for better understanding the fisheries and helping more informed management decisions. And this is a good time for collaborating and tag more fish because in the Mediterranean Sea, we don't have a lot of tagging fish. This is important for biodiversity. This is also a larger ambition to create the European-wide network, and there are many co-producers in the Mediterranean. And now we are in the Greece, in the Lesbos. This is a good time for agency telemetry network because we are close together, and Greece and Turkey need to create the agency telemetry network to track important aquatic animals in the agency. And this is the idea. The Greece and Turkey is a lot of sea cages in the agency. We can start to put some receiver sea cages to track the animals and collaborate in each other. This is a slide also show some sea cages in the Turkey and in the Greece. The blue one is Greece farms. The red one is a Turkey farms. We can put some receiver in the cages, and we can tracking any animals around the cages using the monitoring station. Sometimes, no? Please continue. Okay, sometimes it is. A great Turkish researcher consortium might create research proposal targeting European coasts. We can get support from the ETN to find a budget or training for the researchers. And also finally attend the ETN meeting, next ETN meeting in the this October in Mallorca, get their support or funding applications. Thank you for listening, and if any questions, please.
So first, congratulations for your work. A small question. Can you go to the slide of the, I think it was the tuna migration pattern? You used both labeling and acoustic techniques or just the acoustic techniques there?
The migration of the tuna is not acoustic method. It's a PSAT method, but some researchers in the Atlantic, they put a lot of receivers in the Atlantic coast, and they also take the bluefin tuna with acoustic transmitters, and they understand migration now. And they get a detection from around to also Mallorca. If we can take any bluefin tuna in the agency or in the Met, we can also get a detection from the acoustic receivers. This is the idea why we put some receivers also.
So you need the combination of tagging and
yes,
acoustic
double
techniques.
tagging, the PSAT tagging, and also we need the acoustic tagging.
Okay, okay,
yes.
thank you.
But it's cheapest method comparing than PSAT.
Mm-hmm.
It depends of the environmental conditions, because the question is not easy to reply. For example, in the agency in HydroMediT Bay, we get a detection almost 600 meters, sometimes 700 meters, but in the darkness, is it sometimes 200 and 250 meters. Depends on the sea traffic and environmental conditions. Because the receiver only listens to noise, the transmitters. If a lot of noise, the receivers don't hear the transmitter pings, the basic idea. But in the agency, if don't you work in the channel or like this or in harbor, the detection range is very nice, more than 500 meters. The marine mammals is very difficult because of the ethnic rules, but some researchers used also sea turtles, put some epoxy on the shell, and then they also track easily.
I'm Evrim Kurtay, and we are working together with Aytaç Özgül. And I want to add something about the range of the receivers. So the new model of it's on selling now, in market now, the next generation. Yeah, they have a new model that hears from far distances, like 1 kilometer away or something. Yes, and the technology is 1.5 actually, and the technology is developing every day. So it will be useful to listen under Stecken Reuter's seminar next after this session. I don't know. I'm not ready. Yeah, 1 o'clock. 1 o'clock. Yes. Yeah. Thank you.
And I want to call in the podium Mr. Varkoulis, who will present the preliminary results on myofunnel community structure of different habitat types of the Mediterranean Sea.
Hello, my name is Varkoulis Anastasios, and today I will talk to you about some preliminary results regarding the myofunnel community structures of different habitat types of the Mediterranean Sea. Here we define habitat types as the terms given in the framework for conservation practices throughout the Mediterranean, which was arranged in the Barcelona Convention in 1995. Among other things, it includes descriptions for habitats and biocenosis, and the most important features for these descriptions are bathymetric position and the type of the substrate. The habitats of interest are firstly the soft substrates, which vary with grain size homogeneity. They cover vast areas and are considered reference habitats mainly because they balance out certain stress factors which can be seen in other habitats, nearby habitats. Then we have angiosperm meadows and especially meadows of Posidonia oceanica. Posidonia oceanica is the most common seagrass of the Mediterranean Sea. It is very sensitive to environmental stress, and lately we have seen it being replaced by other angiosperms and even invasive species. And finally, we have some faunal associations like bivalves, corals, and sponges. These phases of fauna can sustain great biodiversity, and it is also documented that they can alter the nutrient cycle of the ecosystem they can be found in. Now some words about the meiofauna. Regarding their size, meiofauna are between the microfauna and the macrofauna. They are considered key players in energy flow of several ecosystems from the deep sea to shallow waters. And they can also be used as bioindicators as they are very sensitive to abiotic changes. They present very high beta diversity among different parts of the same habitat. For example, the stem of seagrass and the rhizome of the seagrass. And they can occupy biogenic habitats, as I said, like seagrass leaves and also other such types. The scope of the present study was to describe the meiofaunal communities in 4 different sediment habitats. First, we have Posidonia oceanica meadows. Then we have well-sorted sandy sediments, Zostera marina meadows, and finally sediments adjacent to Pinna nobilis, the largest bivalve in the Mediterranean, which is also going extinct nowadays. I will now briefly explain the sampling design we followed. We conducted seasonal samplings in one locality west from the Gulf of Volos, which is located in the central Aegean. We collected 3 replicate corers by scuba diving. Overall, we collected 48 samples. And here you can see that Posidonia oceanica was at the deepest part of the location, and the well-sorted sand was at the shallowest. I should also mention here that we had 2 vertical zones of the sediment. The first 5 centimeters were taken and analyzed, and the results are presented here, but we also have sampled the latter 5 centimeters. For the extraction of the meiofauna, we sieved the bulk of the sample through a 500 micrometer sieve, and the meiofauna were retained in a 63 micrometer sieve. Then we used the flotation method with Ludox, which is based on the specific density of the liquid, which is higher than that of the meiofauna, and thus the organisms flow on the surface. We then stained them with rosa bengal, and finally we counted and sorted them to major taxa under a stereo microscope at 40 times magnitude. For estimating the diversity, we used Hill's numbers. Q equals 0 means the species richness. Q equals 1 corresponds to the Shannon diversity. Finally, Q equals 2 refers to the Simpson diversity.
Then we constructed the air fraction extrapolation curves through the INX package of RStudio. And to compare the habitat types, we used hierarchical clustering with Ward's method. And finally, to find the species or the taxa responsible for these differences, we used the simple analysis. So the results show that well-sorted sand presented the lowest species richness. It also had significantly different counts of frequent and dominant species. This is the Q1 and the Q2 numbers. Also, Zostera marina and Pinna nobilis exhibited the highest myofaunal densities, approximately 10,000 and 500 respectively. Hierarchical clustering resulted in 3 clusters. Zostera marina and Pinna nobilis will cluster together, and it is also noticeable that the seasons of each habitat were clustered together with the exception of Posidonia oceanica in autumn. The simple analysis showed that nematodes contributed in all comparisons. They distinguished all habitat types, except for well-sorted sand and Posidonia oceanica. Then we have Celiophora, which contributed to the differences between Posidonia oceanica with sand and Pinna nobilis. Oligoheads differentiated Zostera marina and Posidonia oceanica, and finally polyheads and copypods differentiated sand, well-sorted sand from Posidonia oceanica. From these results, we conclude that that Pinna nobilis and Zostera marina can be considered one myofaunal community. We can also see that the effect of seagrass species is evident. We saw that Zostera marina was significantly different from Posidonia oceanica. This contradicts with previous studies on vegetated versus unvegetated habitats, which showed that vegetated habitats always had higher myofaunal abundance. We didn't see that here. Zostera marina indeed had higher myofaunal abundance, but this was not the case for Posidonia oceanica. In the future, we will use nematode communities to describe these habitat types. We believe that because nematodes are hyperdiverse, we will even find some species that are strictly found in one of these habitats. And this study showcases that the inclusion of myofauna in future studies will help regarding conservation policies, as myofauna can adequately describe the identity of its habitat type. Thank you for your attention.
Thank you for your presentation. Yeah, thank you. I'm Ozan Soykan from Turkey, from Ege University. My question is, especially in Turkish coasts, we lost vast majority of the Pinna populations due to an illness. I would like to know what is the status of the Greek coasts regarding the Pinna population. Do you have any observation?
I'm not sure, but I believe that over 90% is extinct right now.
Yeah.
So some of the cells that we used were dead cells.
Mm-hmm. So more or less it's the same.
Yes.
Good morning. Nice presentation. Dros Kotsoubaras, Professor of Marine Biology in the Department of Marine Science here at the University of Aegean. Just to complete your question, because our department, the Marine Biodiversity Laboratory, has performed such investigation, we face the same problem. more than 95% of the lived population have been gone away, have been extinct because of the infection, mostly by Aplosporidium pinnae, but also Mycobacterium and other. So yes, the same situation. However, we should mention that it seems that there are particular areas, probably like in your country, in the Sea of Marmara, or here in the island of Lesbos, where we have the Kaloni Gulf, and also Avrakios Gulf in the western part of Greece, where we still find alive specimens of Pinna nobilis because the situation there, the high dynamism of this kind of transitional water ecosystems make the microbes not to live there. So the Pinna nobilis population is still thriving there, but only some exceptions. Also Sea of Marmara. Following the question of my colleague here from Turkey at the university, the question is, I think you answered earlier that actually when you did the sampling, you didn't sample from areas, the Pinna nobilis you're referred to is not alive, right?
Most of it is not alive, yes. There were some individuals that were alive, but most of them were empty cells.
Okay, and when another question is, when you refer that you have sampled Pinna nobilis habitats, is it something different speaking? Because obviously, if I understood well at least, you haven't sampled from the animals, the cells of the animals itself, themselves, sorry. So you have sampled sea bottom where Pinna nobilis, dead or alive, mostly dead, were found, right?
Yes.
What's the difference of this habitat with the well-sorted sand? Yes.
It was abundance-wise, it was much higher. The numbers of Myofauna were much higher in the sentiment adjusted to Pinna nobilis.
Yeah, that's what I'm asking actually. Is there any difference between the habitat type where Pinna nobilis is on the bottom with a fine sorted, well-sorted fine sand? Is it a different habitat? That's what I'm asking actually.
There is, we could say that it is a different habitat because on the cells of the dead Pinna nobilis, we found certain macrofauna species. So the sediment itself was different from the bare sediment, let's say.
Okay, thanks a lot.
Thank you.
And now we're welcoming Mr. Vulgaris, and he will talk about mapping myofaunal patterns in semi-enclosed basin of Eastern Mediterranean in Pagasetikos Gulf in Aegean Sea.
Hello, my name is Vulgaris Konstantinos, and today I'm going to talk to you about mapping myofaunal patterns in a semi-enclosed basin of Eastern Mediterranean in Pagasetikos Gulf. So what is myofauna? Myofauna are organisms smaller than 1 millimeter and larger than 0.06 millimeters. We find these organisms in high abundances and they have no planktonic phase. Some other important characteristics are the brief life cycles, asynchronous reproduction, high turnover rates, and the sensitivity to environmental changes. All these characteristics make the myofaunal organisms ideal bioindicators. Our research took place in the mesophotic zone, a zone with a minimum depth of 30 meters and maximum depth around 150 meters, which is an understudied zone in the matter of myobenthic communities. The study area was Pagasetikos Gulf, a shallow and semi-enclosed basin with an average depth of 69 meters, located in the central Aegean Sea. Some important aspects of the Gulf are the intense urbanization and environmental pressure, with a recent example of Daniel and Elias weather events that struck the whole region of Thessaly, and Pagasetikos Gulf in September 2023. Another important characteristic is the limited communication of the Gulf with the open sea. Samplings took place during August 2022 at stations arranged in a grid covering the whole Pagasetikos Gulf, with a 3-kilometer distance between points. Myofaunal samples were collected at each station at a 10-centimeter sediment depth using a Van Veen grab, and subsamples were taken with the help of a 10 square centimeter corer. Depth was recorded with the use of a CTD instrument. At the laboratory, all myofaunal organisms were retained between a 500 and 45 micrometer sieves and were extracted with the use of the flotation method. After the extraction process, all organisms were stained with rose bengal and counted and sorted into higher taxa. All analyses were done with the use of RStudio. To examine the influence of depth on the total myofaunal abundance, a linear regression was conducted. A non-metric multidimensional scaling analysis combined with depth contour lines performed using a Gaussian error distribution, and the permutation analysis of variance were used in order to understand differences between regions of the Pagasetikos Gulf in terms of community structure. Heat maps for the total myofaunal, nematode, and copypod abundances were constructed in QGIS using the function kernel density estimation. The effective radius of each station was determined by the Hooft method. Our results showed 90 myofaunal taxa were identified in all stations. Nematodes were always present and dominating the stations. Ciliates dominated 5 specific stations. All of them were found in depths larger than 70 meters. And copypods was the third most abundant taxon. In picture A, the demonstration of the weak influence of depth on total myofaunal abundance. We can see this. In picture B, we can see the separation of the myobenthic community of the western part with blue color, and the rest central with red and eastern with green, for the Pagasetikos Gulf in terms of community structure. Variation in depth correlates with changes in myofaunal communities along the coast. In picture A, total myofauna, and in picture B, nematode abundance seem to increase from east to west in the Gulf. And in picture C, the copypod density was homogeneous except for one western station. Based on the results, we conclude that the specific hydrodynamic conditions of the Gulf, as indicated by the arrows showing water renewal and current flow, may contribute to increased myofaunal densities. This suggests regional differences between the western part and the other areas of the Gulf. Additionally, the shallow depth stations found exclusively in the western part of the Gulf exhibited lower myofaunal densities, indicating a potential indirect relationship between depth and myofaunal densities. Finally, anthropogenic activities also appear to influence myofaunal patterns, with a notable increase in copypod abundance in the southwestern part of the Gulf, where an aquaculture unit is located. The next step of our research is to examine how the abiotic factors related to sediment and the water column in order to gain a clearer understanding of this shallow and semi-enclosed basin, and this will provide us with insights on how myofaunal communities work on those specific systems. Thank you for your attention.
Okay, thanks a lot for your presentation and for your business. Okay, see you in the next sessions.
Gel seninkini yapalım önce. Tamam. Ver. Ver. Hangisi PPT değil mi? Başka bir şey alacağım mı? Olduğu için şöyle masa üstüne atıyorum ben. Tamam. Nereye attı? Şurada. Tamam mı? Şeyi ayarlayalım. Noktaları. Ama orada gözüküyor. Şöyle bunun mesela şey yaptığında bir dakika.
Good evening to everybody. I would like to welcome you here from the chair. I'm Professor Rania Gioraki from the Marine Science Department. Okay, so we will start this session in the fifth field regarding inland aquatic ecology and resources. We would like to invite the first talker, SEC, and the topic is diet and food selectivity of Aphanius aimeriensis under extreme habitat conditions. So, please.
Good afternoon, everyone. I'm Gülşah Saç from Istanbul University Biology Department in Turkey. I will talk about the diet and food selectivity of Aphanius aimeriensis under extreme habitat conditions. Aphanius aimeriensis is a fish recently described from Greece in 2007. It was considered under Aphanius fasciatus, but the last molecular studies showed that these species are separate species. It's morphologically very similar to Aphanius fasciatus. They both live in Mediterranean basin. Please look at the map. Red dots show the distribution of Aphanius aimeriensis, and the black dots show the distribution of Aphanius fasciatus. Aphanius aimeriensis is more common to Aegean basin, and Dr. Sevan Adamar will present you the details about this fish in his study. It was also recorded from Italy, from a coastal cenote in 2019. This fish, like the other killifish, is an extremophile fish species. It mostly lives in hypersaline waters or high temperature waters. It was recorded from some coastal streams, springs, estuaries, or lagoons. This is our study area, Tuzla Spring. It's a very shallow water. Actually, it was exciting to see a fish living here. It is located in western Anatolia, in Çanakkale province. It's a thermal area. The water comes from the foot of Tuzla mountain, and the geothermal evaporation causes the spring water to reach a temperature of around 70 centigrade degree, and the spring connects to the Aegean Sea by Tuzla Creek. There are both cold and hot water outlets in the area. So the area is a wet area, and maybe you can see here some salt formations on the rocks. All the filtered waters, hot or cold, comes here and mix here, and there's a road, and the water, spring water, flows through a funnel here, and then it's after the road, it consists a swamp area, and then it connects here from here to the Tuzla Creek. Maybe you can see some fish samples here. It's a very shallow spring. It has extreme habitat conditions in terms of physicochemical parameters, firstly, high water temperature and salinity. And water quantity is variable here. Since the spring is fed by both rainfall and groundwater, there's always water here in the spring. But the amount of the water varies depending on season. We could observe it, but we couldn't measure because yes, there is always water, but in the swamp area, it have a marshy bed, so it is difficult to measure the water depth. So we collected the fish samples from the first mixed area here. The field surveys were conducted during 4 seasons from January to October in 2023. Actually, we have wanted to do sampling, multi-samplings, but we avoid hunting pressure on these small populations. Fish samples were collected using a scope net with a mesh size of 3 mm, you can see here. And aquatic macroinvertebrate samples were collected using a special hand net with a mesh size of 0.5 within a 1 meter square quadrat area. And then we also measured some water parameters, physicochemical parameters, such as water temperature, salinity, dissolved oxygen, pH, TC, and conductivity. You can see the seasonal variation of physicochemical parameters of the water in the spring. All values changed seasonally as expected, but especially water temperature and salinity was very high, especially winter and summer. Winter salinity in winter and water temperature in summer. We had some questions before the study because it was very interesting area, and this fish have survived in the area. So these are some of our questions. What is the diet and niche breath of Alphanus almeirensis under the extreme habitat conditions of Tuzla Spring? And the second one is, are there any temporal differences in the feeding of this fish? Firstly, a total of 120 fish samples were examined, 30 per seasons. The digestive tracts were removed from fish samples, and the prey items were categorized to the lowest possible taxonomic level using both binocular and microscope. And then especially countable food items such as insect groups, maybe you can see here Edipteran samples, were counted. And then all types of food were dried at 18 centigrade degree for 2 or 4 hours, and then waited. To answer these questions, we used some indices. The first one is vacuity index. It was a measure of feeding intensity of for the fish. And relative importance index and modified index of relative importance. These two indexes were used to find the importance of each food groups in the diet. Maybe you can see differences of these two indices. Relative importance index uses the M values. I mean, it was estimated for countable food groups. And the other one is used only weight or frequency of occurrence of each food groups. And the Lewis index was used to determine the niche breed of the population. And we look at the results, we showed that the vacuity index, a measure of feeding density, was 0 for all seasons. That means fish always had something to eat. And these are the food groups found in the digestive tracts of the fish, of the populations. Insecta was consist from Diptera, Coleoptera, and also unidentified body parts, some dipterans or insects, we don't know, some legs or, and also insect eggs. And these are diatoms, Navicula, Nitzschia, and Holanphora. Maybe you can see in this picture, in this photograph. And also cyanobacteria for medium, and also cocoid cyanobacteria here. Annelid, arachnida, nematoda, and also fish we have found. You can see the fish scales or fish bones in here, and plant debris. Actually, arachnida is not an aquatic organism. It's probably a drift organism, and fish caught it. Here you can see the seasonal values of frequency of occurrence, relative importance index, and also modified index of relative importance. Here the asterisk indicates the unidentified taxa. I mean, some body parts of dipterans or insecta. The highest frequency of occurrence values, F%, were for diatom and cocoid cyanobacteria for all seasons. And you can see them in red. We found these two types of foods in the guts of every individual examined in all seasons. And the relative importance index, it was estimated for only countable insect groups. It was high for AMPHITHEATER in winter, HydroMediT in spring, and Ceratophragonidae in summer and autumn. And the last index, a modified index of relative importance, was estimated as high for diatom and cocoid cyanobacteria in winter and spring. It must be purple, but I forget it. And for medium, for summer and autumn. Here also, I have also summarized the results. Diatoms, cocoid cyanobacteria, and plant debris were found in the digestive tract of each individual in all seasons. Aphanus almeriensis feeds intensively on diatoms and cyanobacteria, which are particularly easy to catch and eat throughout the year. And in summer and autumn, food diversity decreased, but for medium showed dominance with very high values, more than 8%, in terms of modified index of relative importance. And among the identifiable insect groups, AMPHITHEATER has a high importance value in winter, while HydroMediT and Ceratophragonidae dominate in all other seasons. P-value, it means Levin's niche index for the population was estimated to be 6.7 or 8 with a narrow niche breadth. It's relatively low for the fish that appear to consume a total of 16 different food items. And the species consumes a variety of foods, but the dominance feeding on some food items such as diatoms, cyanobacteria, or formidum, this index was resulted in a low niche breadth value. And you can see the differences between the seasons in niche breadth. It was low for winter as to be 4.3. Here, the population has consumed 12 different food groups or prey but their occurrence, you can see here the values, was very low, except diatom and cocoid cyanobacteria. However, the value was estimated high in spring, and the occurrence of other food groups has increased in these seasons. So the B value has increased. And the other question of the study, what is the food selectivity of fish in its extreme habitat, and how is it changed seasonally? To answer this question, we have also sampled macroinvertebrates in the environment, and then identified using binocular or microscope and their relative abundance of each food items, as actually each prey macroinvertebrates in the environment was estimated using this formula. And then to assess the prey preference of the fish, we estimate Ewley's selectivity index for selectivity. Here you can see the process. Macroinvertebrates were collected from the spring, and then they were fixed in formaldehyde solution, and then they were washed under at tap water, and they were identified using microscopes, and these are some groups: HydroMediT, Ceratophrogonidae, Amphitheridae, and Hirudinea. And extreme habitat conditions indicate that the environment is poor in terms of macroinvertebrate diversity. Only 7 taxa with a total of 124 individuals were identified. And Amphitheridae, you can see the differences between seasons. And Amphitheridae were sampled in all seasons and were more abundant than the other taxa in terms of number of individuals during the year. In winter, only 3 different prey was identified. Amphitheridae more abundant, and spring only Amphitheridae and Coleoptera. And summer and autumn, only dipterans were found in the environment. And here you can see the electivity index results for each season. In winter, Phanius was not selective for the foods or prey found in the environment. You can see environment, Amphitheridae, Hirudinea, and Gastropoda we have found, but the selectivity was minus, estimated as minus. Amphitheridae, yes, fish has consumed Amphitheridae, but it wasn't selective. It wasn't positive selective because its volume in the environment was very high than the fish guts. And in spring, it was positive selective for HydroMediT and Ceratophrogonidae because we didn't find them in the environment, only in fish guts. And in summer and autumn, the selectivity index was estimated positive for HydroMediT and also Amphitheridae in autumn. 3 different macroinvertebrates groups were sampled from Tuzla spring. In winter, the fish consumed only Amphitheridae individuals and were negatively selective for them. Although HydroMediTs, coleopterans, or annelids were not obtained in the environment, they were consumed by the fish in low numbers, resulting in a positive selectivity. Only Coleoptera and Amphitheridae were sampled in spring, and although the fish consumed them as food, they didn't show positive selectivity of these groups. And in summer and autumn samplings, the environment was represented by only 3 different groups of Diptera, while the fish were positively selected for HydroMediT in both seasons and for AMPHITHEATER in autumn. Although ceratophagonidae were found in the environment and the fish died in both summer and autumn, the selectivity was negative. This can be explained by the higher abundance of this prey in the environment than the fish did. In conclusion, Aphanus almeriensis lives in the extreme conditions of the Tuzla thermal spring in terms of high salinity and high water temperature. To survive, this fish is an omnivore. Its diet consists of both plant and animal organisms. This fish feeds main known diatoms, cyanobacteria, and some insect groups, especially dipterans. The importance or frequency of food items varies seasonally. Although 16 different foods were found in the diet of the fish, the niche breed is moderate, as the species feeds predominantly on some groups, as I told you before, diatoms, cyanobacteria, or formidium. And the habitat of the fish is poor in macroinvertebrates, and the fish shows seasonal selectivity for some different macroinvertebrates. However, it's particularly selective for HydroMediT in all seasons. Thank you. This is my team, and they are my team. We all thank you for your attention.
Interesting presentation. Do we have any questions from the audience?
Gülşah Hocam çok teşekkür ediyorum. Benim sorum sen iki görevin var. Türkçe, önce İngilizce çevir, sonra sorumu cevaplayacaksın. Okay? Şimdi çok güzel bir çalışma. Çünkü bu küçük balıkları ki herkes göz ardı eder. Bu nedenle yapmış olduğunuz çalışma çok değerli. Örneklemelerde özellikle dişiler mutlaka açtınız. Yumurta durumu nasıl? Sanırım o çalışmayı başka zaman vereceksiniz. Üremeyle ilgili çalışma verecek misiniz? Peki yumurta durumu nasıldı? Bir de balıkların buradaki popülasyonu etkileyen çevresel koşullar nelerdi?
Okay. Okay. He asked the fish eggs reproductive capacity. Actually, we didn't count the eggs because fish was very, very small, and there was too many small oocysts, and we didn't estimate fecundity. And also, we just sampled only 1 month in spring. So we have nothing to say its reproduction. İkinci soru neydi hocam? Second question. What was the second question?
İkinci şey mi? Çevresel koşullar nasıldı? Popülasyonun etkileyen çevresel koşullar nasıldı?
The environmental conditions was actually horrible because it's. Let's look at. Can I?
High temperatures.
Yeah. Both hot and cold waters mixes here, and water temperature was very high, 14 degrees. And also, it is very open area, human pressure, and also there's a road, macroplastic pollution maybe. There was very plastic bottles around here. So we have to protect this population. It's very small population. Environmental pressure very high.
Is very, very easy. And I wonder, is there any ponds there?
Other side of the boat is the swamp area. A pond, but very marshy. So fish found especially here, and this
on the drinking water when you have flow. flowing water or on the ponds?
Flowing water.
Flowing water.
It was connected to agency by Tuzla Creek. And there's a small, you can see here, a small canal. It was then connected to this Tuzla Creek and then agency.
Other questions? Or from the internet, is there any chat? Please write if you have any questions.
There's no question.
No questions. I would like also to ask you, is this a protected species, this Aphanius fish? Protected species?
Protected species. But I have to say that female was dominant in the population. More than 90% was female. They are productive here. They are üremek. They can reproduce here. Sorry, thank you. Reproduce here, and also females dominate.
Well, thank you very much. Because
I
we
thank
have
you.
more time for more questions. It's very interesting work.
Thank you. Thank you very much.
I'm
very
see
excited.
that
Sorry.
published and in comparison with the rest areas that you have shown us to the Mediterranean. So we move to the next presentation.
Thank you.
Hello, I am Gözde Şavran from Sparta University. I will tell you about the first data on the biology of the Semnomachilus spartansis in Anatolia, endemic species. Turkey shows different characteristics in terms of altitude, precipitation, temperature, topography, and geology. These differences create These differences create habitat and species diversity. We can clearly see this diversity in inland water fish. Considering the last researches, there are around 4,030 inland water fish species in Turkey. Of these, 215 are endemic species. In other words, 50% of the inland water fish fauna in Turkey consists of endemic fish. When we compare the species diversity of inland water fish in Anatolia and Europe, while 441 species are found in Europe, 420 species are distributed in Anatolia. When we look at the IUCN studies of fish in Turkey, there have been 4 types for eggs and 32 types critical categories. Additionally, approximately 17 species eggs candidates. Semnomachilus genus from the Nemechilae family, distributed in the inland waters of Turkey, is represented by 6 species. Semnomachilus spartansis, which is the subject of the study, is distributed only in the Eğirdir Lake Basin and Beşevler stream, which is a closed basin. In our study, we studied the endemic Seminamachilus spartansis populations exposed to these effects in Lake Erie. Sampling was carried out seasonally with a total of 79 samples with multi-mesh clinics according to modified TSE and 40,747 and beach say net in the study area. Age range, male-female ratios, male and weight range, length-weight relationship, both Bertalanffy growth equations, egg number diameters, and fecundity were determined. Endemic spartansis age range in Lake Erie 2-4 varies between. Dominant age group, 2 age group, 46.74% of the fixed examined in spartansis populations of Lake Erie is male and 43.05% are female individuals. Spartansis late at wake range in Lake Erie are 7.39 to 20.13 centimeters, 4.14 to 22.16 grams. The land weight relationship was calculated and the mean value of the condition factor was calculated. Condition factor 1.10. The exponents of the total length weight relationships of spartansis show positive allometry. Other Seminamachilus species, positive allometry growth was generally observed. You're seeing the regression coefficient R square value is generally similar to other studies. The von Bertalanffy growth equations of endemic spartansis populations, asymptotic length, populations is 17.45. Difference between observed and expected total lengths were found to be statistically significant in all age groups. The average growth performance was calculated as 1.09. The differences between the observed and expected total lengths of spartansis were found to be statistically significant. The population is predicted to reach a length of 17.45 centimeters, asymptotic length at approximately age 30. Since there is no study with asymptotic length that regarding the species, the result of our study could not be compared. Spawning occurred between April and July. The GCI values reached a maximum in April and declined rapidly thereafter, reaching a minimum in July. In our study, the result we obtained regarding the reproductive characteristic of the species constitute the first data of the species. There is no study other than our study. Considering the state of the species habitat, flow is low, the AXU, hydroelectric central, kaya ağzı, partially dried, complete dried, karadut, boyalı, complete dried. Marble quarries in the feeding basin of the lake, Köprübaşı location, partially dried. Apple gardens reaching up to the lake shoreline and wetland turning into pasture. A wetland turning into pasture. Aidra nigra habitat, complete dried. Habitat loss. According to IPCC climate report, it has been reported that the Mediterranean region, including Turkey, will be one of the most prominent and sensitive points of climate change. It is estimated that surface temperatures in the region will increase by at least 3.5 degrees by the end of this century, and summer warming will be at least 50% greater than the global increase. Since decades where our study was conducted is in the Mediterranean region, it has been affected by this situation. Its water volume has decreased significantly, and many wetlands around it have disappeared and turned into pasture. Due to the decrease in the depth of the lake, it is seen that the process that causes the natural structure of the lake to deteriorate has begun. Adverse conditions may arise due to the intense proliferation of aquatic plants. It was evaluated that taxa belonging to cyanobacteria were relatively dominant in terms of quality and quantity. This can create problems for water quality and aquatic organisms. Although the population data of the Spartansis species, which is the subject of our study, is generally positive for now, we think that the population size will be in danger in the near future due to the significant decrease in habitat areas and the pressure of invasive species in the lake. For this reason, conservation plans that have been made need to be taken over. Let's protect our wetlands. Thank you. Thank you.
We move with the next presentation.
Hello, dear chairs and participants. My name is Sevan Adamak. I'm a researcher at the Department of Forestry, Çanakkale 18 Mart University. Today, I will try to represent you about our study named First Molecular Description of the Validity of Aphanius armiriensis from an Extreme Freshwater Habitat in Turkey. Dr. Yusha Saç gave you information about the feeding ecology of the species, and I will briefly give you information on the molecular identification of the species. The small killifish Aphanius armeniensis inhabits from brackish springs to estuaries and coastal lagoons in the eastern Mediterranean basin. Also in recent years, molecular-based studies have led to a much more restricted definition, limits, and contents for the genus Aphanius, which previously belonged to the family Spinodontidae. This small-bodied fish, Aphanius armeniensis, which is very similar to Aphanius fasciatus, known from the Mediterranean coast, was described as a new species from the Peloponnese region by Koterat et al. And immediately after Trinthiophyllidis et al. Found two well-separated molecular groups in Greek Aphanius fasciatus. It is very important to understand evolutionary mechanisms that cause species living in fresh, brackish, or hypersaline water bodies to differ from one another. It is only through phenotypic and physiological adaptations that fish can cope with extreme salinity or temperature conditions in water bodies. Our study aims to identify Aphanius species inhabiting the Tuzla thermal spring, which exhibits a narrow habitat specificity. If you look at materials and methods, Tuzla Thermal Spring is a shallow salinity-rich water from the Biga Peninsula in the southwestern Turkey. The study area was visited in January 2023, and fish samples were obtained using a scope net with a mesh size of 3 mm. The collected specimens were cured with an overdose of chlorine oil and stored in ethanol at minus 20 centigrade degree. Water temperature and salinity were measured using a multiparameter. Genomic DNA was extracted from cow dolphin tissue using Kaijin DNA Easy Blood and Tissue Kit. Primer pairs were designed to amplify only the DNA of target species from tissue content. DNA amplifications were performed in 25 microliter volumes, each containing following. And PCR amplifications were performed in append of master cycle with the following cycling conditions. And also Sanger sequencing was performed by BM Labosis with reverse primer. Molecular data analysis of Stockholm oxidase were conducted from 10 specimens from Aphanius. Nucleotide sequences were aligned using cluster W algorithm in MEGA10. DNA sequence data were submitted to GenBank and the accession numbers were assigned. Molecular taxonomic cladogram was generated in MEGA10 using maximum likelihood algorithm with 1,000 bootstrap replications. Genetic distances between species were calculated in MEGA10 using the P distance model with 1,000 bootstrap replications. Reference sequences of species on cladogram were obtained from GenBank, and you can see the table 1. If you look at results, figure 2 shows a molecular taxonomic cladogram based on the DNA fragment of the mitochondrial Stockholm oxidase gene from 10 specimens of Aphanius armeniensis, and its closest congenic species showing the monophyly of all of them with good statistical support. The conglomerate was constructed with high bootstrap values based on the maximum likelihood analysis of Stockholm oxidase data, and the tree suggests that Aphanius armeniensis specimens from Çanakkale were clearly distinguished from all other Aphanius species obtained from GenBank. 10 Stockholm oxidase data obtained from Aphanius specimens are similar to each other and cluster independently of those of the taxonomically closest Aphanius species with genetic distance values typical of an independent species. Salinity and temperature values of the Tuzla thermal spring were measured at high levels with 61 ppt and 31 centigrade degree, 34 centigrade degree, respectively in winter conditions. Also, it is important to note that the salinity and temperature values obtained are higher than those of seawater, approximately 38 per 1,000 in the Mediterranean Sea, showing that the fish have adapted to extreme conditions. Concerning all the above data, this study is the first molecular record of Aphanus armoriensis from Turkish inland waters. And further studies could focus on the genetic mechanisms of the species adaptation in these extreme conditions. And thank you for your attention.
Question? Also from the internet, I don't see any. Yeah. So what do you think is the main difference that you find in the genetic material actually for this population from the other areas in Greece or that you can make? What is the main difference?
Not kind of main difference, but only geographical differences, and also this species looks like Aphanus fasciatus, but these extreme conditions make some change in genetic
Okay, it's genetic material.
Yes,
so you think that its genetic material has been altered and was adapted to this difficult saline conditions?
Yes.
Not only saline, but in general high temperatures and difficult conditions. Thank you. So I think there is much work into that still to understand how they are adapted. The molecular material of the organisms, they are adapted in the difficult environments. Thank you very much. We move forward to the next speaker.
Welcome. Thank you.
So, hello from me. Starting, right? So, my name is Maria Alvanou. I'm a PhD student in the University of Western Macedonia. And we are from the Laboratory of Ichthyology and Fisheries from Aristotle University of Thessaloniki and from University of Western Macedonia, of course. So today I'm going to discuss a really important topic regarding the mass mortality events of Pondostachys leptodactylus populations in two lakes in northern Greece, in Lake Polyphytou and Lake Vegoritida. Not working, sorry. Okay. I don't know if you're familiar with this species. I think you are because also it has a wide distribution in Turkey. So Pondostachys leptodactylus is a freshwater crayfish species, and it has both high environmental and cultural significance, but at the same time, it's really important for biodiversity because it operates as a scavenger that leads to ecosystem balance. So our main goal, of course, is to develop an aquaculture protocol for this species, and both for its high economic value, but as for the restocking purpose as well. Now, what from our current publication, we can see here the distribution range of the species in Greece. Its native range, it's in Evros region. However, during last years, many humanly translocation took place,
And we can see that the new records are from Lake Volvi and Lake Vegoritida as well. In Lake Vegoritida, it's also the mass mortality events that took place. So a little bit about the pathogen. The pathogen called the Phanomyces stasi, it's Noomycetes. It's categorized among the 100 worst invasive species. And what it happens, it's that it penetrates through crayfish cuticles, and then it causes melanized spots, and the crayfish can't walk, and after some time, it's dead. So, what was the problem? The problem, as I mentioned already, was the massive crayfish population reductions in both lakes, Polyphyta and Vegoritida. So we wanted to investigate what happened there, what caused these mass mortality events. And one possible reason for these mortality events is this pathogen, as it's already known that causes mass mortality events in other European countries. So what we did actually, we collected samples from both lakes, from Lake Polyphytu and Lake Vegoritida. And we noticed this spot, this melanized spot, as we can see here in the pleopods and in the crayfish cuticle. And also we observed some paralytic behaviors that animals exhibit. So we extracted DNA, and then we conducted a conventional PCR for this pathogen. And with Sanger sequencing, we identified the pathogen for the first time in Greece. So we do believe that, of course, this leads to the mortality events, as it has already been recorded in other European countries, as this species is very vulnerable to this disease. So all of the samples tested were possible to Phanomyces stasi. One interesting thing was that in Kabi, it was referred that in Greece the pathogen was absent. However, also other crayfish species almost extinct from Vegoritida Lake. So this is now updated, and it's really important. And the questions rising here mainly concerns how does this pathogen has been transferred in Greece. What are the parameters that leads to a possible disease outbreak, and if we can do something in order to prevent a future disease outbreak. Now, regarding the first question, how does this pathogen has been transferred? There are some possible scenarios that we try to investigate and to find to lead to a conclusion. However, it's not that simple. As I mentioned, this pathogen is an invasive species, and it was transferred in Europe by its natural carriers, which are some North American crayfish species, such as Passiphastaxus leniusculus. So Passiphastaxus leniusculus, it has established population in Lake Agra since some year ago, and we also collected samples from there. We detected the species, and we know it has established populations. So we tried to investigate if this was a possible way of transfer to the other lakes, as it's very close with, as we can see here. It's not moving. If we can see on the map, the lakes, the Lake Agra and the Lake Vegoritida and Polyphytu are very close to each other. So if this invasive species carries the pathogen, it can be transferred to the other lakes as well, by fishing tools, by other animals, by human actions, of course, and by connected water bodies. So we extracted DNA as well from these samples, from the Passiphastaxus leniusculus samples, and we failed to find an infection. So of course, this does not mean that they are not effective. We just failed at first step to find a positive sample. Okay, the second scenario was that maybe the Pontostachys leptodactylus, it's a latent carrier of the disease, and under some adverse environmental conditions, such as increased stress or increased temperature or increased rainfalls, the animals get stressed, and then the disease outbreak happens and they die. So how can we get one first picture of what happens here? We know that this pathogen has 5 distinct genotype groups. More specifically, I'm not going to refer to all of them. I'm going to refer to the most important ones. More specifically, the genotype A, type A, is solely found in European crayfish species. So the latent carriers of Pontostachys leptodactylus, they can possibly have this genotype group, the genotype group A. And the other scenario is to carry genotype B, which is isolated from signal, or C, which is isolated from signal crayfish lineusculus. Passiphostachys lineusculus. So after investigation, again, we found that it's infected from genotype B. So we think that the most possible scenario is to be transferred by Passiphostachys lineusculus that has established populations at a very close lake. Further, more recently, on March of this year, it was detected another invasive, invasive and non-native, North American species, freshwater crayfish species in Greece, Procambarus clarki, which is also known to be a carrier of the disease. So we need also to investigate that scenario. And what else we want to check is that we want to check the pathogen's presence in water by using environmental DNA technique, eDNA. We also want to develop a non-invasive method, such as environmental DNA method and DNA swab. And also to investigate the intermediate host existence, as we know that after this mortality event, the crayfish in the lakes were not that many. So the pathogen can survive at some intermediate hosts. So we collected water samples, and we were able to detect the pathogen in the water sample, which was really important as it's a non-invasive method. And we can also monitor what happens with the pathogen. We also detected the Aphanomyces stasi with DNA swab on healthy individuals. So this turns us back to the first hypothesis we make regarding the transfer. So we can see that it's a latent carrier as well, as they had no infection signs at first. And we collected also some bivalves from the lakes, and we were not able to find an intermediate host. So what are future plans? We are collecting both water samples, animal samples, and probably we have some other ideas regarding what else we can check in order to find an intermediate host. So we collect the samples in a seasonal pattern from January to December. And also we collect some physicochemical parameters like temperature, oxygen, turbidity, and salinity, as we notice that turbidity, especially, maybe it's correlated with increased stress of the animals that leads to disease outbreak. And of course, we want to see at physiological level of the animals what happens to the level of stress by monitor some HSP, for example, genes to prophenol oxidase system, which is correlated with the disease outbreak, and of course to the oxygen defense of the animals with qPCR. So yes, it's a complicated problem, but thank you very much for your attention.
propose to present it is several scenarios.
Yes.
First of all, you have started to check if there is a transfer, the parasite actually, or from infected crabs. This is a kind of Astacus, you call it.
Yeah, actually, it's called Astacus different genera, but yeah, Astacus is also a freshwater crayfish.
What you are studying, Astacus, you call it.
Yes. Let's say yes. Yes.
So the first scenario that you examined, you thought that maybe it's not the case initially.
Actually, we thought that it was transferred by a non-native crayfish that is inhabiting in a very closed lake. So this is a natural carrier of the disease. So we hypothesized that maybe it was transferred from this other non-native crayfish.
But there wasn't any evidences.
There was, because we genotyped the haplogroup, and we found that the haplogroup is the same that also this non-native crayfish carries. The haplogroup B. But then we also tested some healthy individuals that had no signs of infection of the same species, I mean. And we also noticed that it's a carrier of the disease without even having any signs of infection.
It's also interesting that they are carrier. And you mentioned about stressful conditions that may was the reason that the carriers
lead to the disease outbreak. Yes.
Yes. And did you examine these stressful conditions in these big lakes?
Actually, yeah, we have some preliminary data that we are now trying to analyze, and we collected seasonal data of seasonal samples, from all months during the year, and we're trying to investigate how some stress genes, such as other genes, what happens there with the expression of these genes, both at expression level and protein level as well.
In other lakes in the Mediterranean, where the same phenomenon is observed, what is the most the reason that they can explain this?
This outbreak.
Yeah. Yes.
I think there's nothing. It's not black and white. Yeah, it's not the clear the reason why this disease outbreak happens. I think it's a combination from both physicochemical parameters. For example, turbidity during this outbreak was really increased. We have this data that was when the turbidity was increased, we noticed this disease outbreak. But of course, it's not.
Is it accompanied by dissolved oxygen reduction?
Yes, of course.
And temperature increase already?
Yes.
And
but we need to.
Is it accompanied also by the extinction of other species?
I'm not sure about that, because also this species was translocated to this lake. It's not in the native range. So I'm not sure about that.
Thank you very much.
Thanks. Thank you.
To close the meeting, close it or όχι, Αυτές τις παρουσιάσεις τις μαζεύουν κάπου, μάλλον, ναι.
Test. Good afternoon, ladies and gentlemen. We are ready to start this afternoon session. I have an announcement of the company Innovasi. Outside, there is, from amphitheater, there is a stand of the company, and there is a plastic fish with a transmitter, and you can play with that and to do some things with this fish of this company. So we can start with the first presentation, which died seasonal changes of fatty acids profile in white musk and liver of gillhead sea bream Sparus aurata from Mrs. McCree.
Good evening. I am Vasiliki Makri. I'm a postdoc in Aristotle University of Thessaloniki, and my study is seasonal changes of fatty acid profile in white muscle and liver of Sparus aurata, including some findings of my PhD study. Let me start by saying a few things of the background. Aquaculture, a main sector of economic activity, is threatened by climate change. The increase of temperature due to the climate changes affecting physiological processes of fish and sometimes affected the thermal limits and leads to mortality. At the same time, the growth of global population requires the increase of food production, and that promotes the development of aquaculture. Energy possesses a key role in tolerance to environmental conditions. Temperature considered a main stressor in physiological processes affecting metabolism, storage and consumption of lipids, proteins, and carbohydrates. Also, fish nutrition relies on lipids to provide energy and vital fatty acids. Fish are the main source of long-chain fatty acids for humans' diet, and long-chain fatty acids are synthesized by linolenic fatty acid and alpha-linolenic fatty acids, and that are synthesized by desaturation and elongation by altering by two steps. As I said, aquaculture is threatened by climate change. The impact of climate change to marine ecosystem is still not understood. This study, the aim of this study is to elucidate the seasonal changes in fatty acids of Sparus aurata in relation to temperature. The samplings were carried out in Larimna, in a fish farm in Larimna in Greece. The tissues of white musk and liver were removed and were stored in refrigerator until further analysis. Also, animals received proper care in compliance with the guidelines for care, and also the protocol as well as surgery and sacrifice conditions were approved by the committee. The first step to determine the fatty acids profile was the lipid extraction. Lipid extraction was performed using chloroform and methanol method. The next step was the fatty acid that were methyl-esterified. And this means that one form of fatty acid ester is fatty acid methyl ester, FAME, which is produced by trans-sterifying lipids with methanol. Methyl esters were obtained using hexane, and the fatty acid profile was determined by GC, gas chromatographer with a flame ionization detector. Also, peaks corresponding to FAME were identifying using a standard combination of 37 FAMEs. This is a typical device of a gas chromatographer. And let's speak about the results. A PCA analysis was performed for each tissue. For each tissue, a separate study showed that polyunsaturated fatty acids explained monthly variation in fatty acid allocation in PC1, while saturated fatty acid and monounsaturated fatty acids explain monthly variation in PC2. The first two figures show the saturated fatty acid concentration and monounsaturated fatty acid concentration in white muscle, but the figure on the top displays the recording of temperature during the days of the sampling. The lower temperature was recorded in February, and the higher temperature was recorded in June and July. Also, the recording of temperature was divided into two periods. From September to February is the acclimatization to winter period, and from February to July is the acclimatization in summer period. Saturated fatty acids concentration, and specifically myristic fatty acid in white muscle, increased gradually in summer period, and palmitic acid also increased in summer period, and specifically in June. Also, palmitoleic acid increased in the early summer period, and oleic acid increased in the summer period and in the early winter period. On the other hand, in liver saturated fatty acids, myristic acid also gradually increased in summer period, while palmitic acid increased in winter period, such as oleic acid. Palmitoleic acid increased in summer period. On the other hand, polyunsaturated fatty acid in white muscle and liver show a different profile, and that depends on the type of fatty acids. But the most of the polyunsaturated fatty acids increased in summer period. The tables show the total concentration of saturated fatty acids, monounsaturated fatty acids, and polyunsaturated fatty acids in white muscle and liver. Total saturated fatty acid in white muscle increased in July when recorded the higher temperature, and polyunsaturated fatty acid increased in the end of the winter period. On the other hand, in liver, saturated fatty acids increased in early winter period, also, and monounsaturated fatty acid increased in the same period. On the other hand, polyunsaturated fatty acids increased in the same period with white muscle in the end of the winter period. This study focused on the investigation of fatty acid patterns in two examined tissues on farmed species Sparus aurata in correlation to seasonal temperatures in field. According to the findings, the seasonal allocation of fatty acids, myristic acid, increased gradually in summer in liver and white muscle of Sparus aurata. Also, total saturated fatty acid increased in summer period in white muscle and liver. It's possible that elevated lipogenic activity is the cause of high saturated fatty acid content, and it had been found the metabolic rate increased in high temperatures. One study found that a reduction of monosaturated fatty acids in the liver of this species of fish due to the liver's role in the fatty acid synthesis and distribution to other tissues, monounsaturated fatty acids in the liver have dropped. Also, due to the same role of the liver, monounsaturated fatty acids levels in the liver decreased sharply from September to December. However, polyunsaturated fatty acids significantly increased in the liver during winter acclimatization, indicating that the fatty acids increased for maintenance during the cold season. Also, the saturated fatty acid content changes favorably with water temperature. However, the polyunsaturated fatty acid concentration fluctuates inversely with temperature. During the summer, Sparus aurata focused mostly on growth and was observed increase of metabolic rate. Also, in early winter, Sparus aurata were devoted more efforts to storing fat. To conclude, pre-winter fish circumstances and seasonal variation in tissue-specific fatty acid storage are significant aspects to take into account in order to comprehend Sparus aurata's capability for overwintering. And also the increase or the decrease of fatty acid or the total fatty acids is tissue-specific and depends on the type of the fatty acids. Thank you.
Thank you, Dr. Macri, for your interesting presentation. Any question from the audience? Okay, I would ask, I know that it was not the goal of this study, but about the concentration of vitellogenin in female Sparus aurata compared with your findings. Do you have any comment about this?
What? Vitellogenin? Vitellogenin, yes, what?
Did you see any differences? I know, but this was not the goal of this study, but if you know about the literature.
No, I didn't see, but
if there is any connection about PUFA.
I didn't examine this, but I have determined the gene expression of the enzymes of fatty acids desaturation and elongation, and we have found a correlation with the activity and expression of these enzymes and the concentration of the fatty acids.
Okay, thank you very much.
Thank you.
Now, the next presentation, entitled "The Effect of Temperature on Growth and Digestive System Autogeny in Amphibian Bisinctus larvae and juveniles. Preliminary results." Mrs. Drakaki.
Good afternoon, ladies and gentlemen. My presentation is going to discuss the effect of temperature on the growth and digestive system ontogeny in Amphiprion bisinctus larvae and juveniles. First of all, let me introduce you to our subject. Amphiprion bisinctus is a species which is commonly known as the Red Sea clownfish. This species is mainly distributed in the Red Sea, and it coexists with 6 species of anemone, and its local habitat is the coral reefs. Clownfish are a particularly popular species with a high commercial value, taking up a large share of marine ornamental species. However, the pressure of overfishing threatens biodiversity, increasing the environmental footprint on the most productive coral reefs. Therefore, larvae rearing is considered the best sustainable solution for both the protection of the environment and to ensure that the increasing global demand for commercial purposes is met. Okay, now we are going to look at the ontogeny. During the first developmental stages, from hatching to juveniles, different morphological and functional alterations are observed in the digestive system. The study of the ontogeny is very important in order to understand the nutritional physiology and to improve the feeding and weaning protocols to promote the development and optimization of the larvae rearing techniques. Temperature is considered a significant exogenous factor affecting the success of larvae rearing, such as the survival rates and the mortality rates. Also, temperature contributes to the optimal performance of rearing protocols and affects the ontogenesis rates of the body systems during the early developmental stages. Indeed, temperature is a primary factor affecting the development of the digestive system. This study was deemed necessary because the number of studies focused on the digestive system ontogeny of clownfish is very limited. Especially, the literature on Amphiprion bisinctus is severely lacking. Clownfish are species with high prospects, and for this reason, further studies should be conducted to ensure appropriate rearing techniques and to develop new feeding protocols. In the present study, different temperature regimes were evaluated in terms of larvae development and ontogeny of the digestive system to determine the most appropriate temperature for rearing conditions. For the experiment, larvae rearing took place at the Quet Aquarium, while the histological analysis was conducted at the Institute of Marine Biology, Biotechnology and Reproduction. Larvae were reared at two separate temperatures, namely 23 and 26 degrees. The first batch consists of 220 larvae, while the second one consists of 55 larvae. The feeding protocol was applied in relation to total length and not in relation to the days of the hatching. Larvae were sampled every 2 or 3 days, from day 0 to day 23 after hatching. Then they were anesthetized and they were measured for total length. The next stage was their preservation for the histological analysis. All the sections were sliced with a microtome and then they were stained following the methylene blue basic fuchsin protocol. Finally, all the sections were examined under a microscope and microphotographs were taken to describe and study the digestive system ontogeny. Now we are going to look at the results of our research. The growth rate of Amphiprion bisinctus is described by linear equations. As we can observe, the slope of the curve demonstrates that the growth rate is visibly higher at 26 degrees compared to 23 degrees. In the following figure, black dots and lines stand for 23 degrees, while red dots and lines stand for 26 degrees. It is obvious that temperature affects the ontogeny of the digestive system, and we can see that most digestive system structures appear earlier at 26 degrees compared to 23 degrees. Also, the lower temperature delays the appearance of some of these structures, including gastric glands, the Y shape of the stomach, etc. However, it seems that there is no difference in total length between the two temperatures. In particular, the ontogeny of the digestive system associated with the larvae total length reveals that most digestive system structures appeared in both rearing batches when fish reached a similar total length. In the following histological sections, we can observe the structures of the digestive system at 23 degrees. I will selectively focus on the function of some of these some of these structures. Sorry. First, the mouth opening on day 0 indicates it marks the beginning of the exogenous feeding phase. And pharyngeal teeth indicate that the larvae started to process live prey mechanically, affecting the successive level of the digestion. In the esophagus area, goblet cells secrete a significant mucus layer, which prevents mechanical injuries, bacterial infections, and facilitates the food ingestion. Okay, now gastric glands, which appeared on day 8 after hatching, is a very important structure because heralding the preparation for the extracellular digestion through the action of the hydrochloric acid and other enzymes. Okay, now we will turn on the liver. The liver is a central organ for metabolism, playing a significant role in lipid and glycogen storage. And finally, the development of pancreas on day 0 is associated with the early ability for the production of pancreatic enzymes. Based on our results, it has been proven that the most appropriate temperature for Amphiprion bisinctus larvae rearing is 26 degrees. Indeed, higher temperature accelerates growth and ontogenetic rates. Similar results have been reached in other research papers, such as Mello 222 for the species Sparsa aurata. The higher levels of ontogeny at 26 degrees compared to 23 degrees enhance the larvae ability to capture, assimilate, and digest feed much more effectively. Without a doubt, Amphiprion bisinctus is a species of high growth rhythms. Also, it's a species which is characterized by high ontogenetic rates of the digestive system structures, as after hatching, a satisfactory digestive system appears. This fact confirms previous studies which state that clownfish are characterized by an increased development of the main digestive system structures. In particular, the rapid development of the digestive tract, the eyes, and functional jaws indicates the larvae ability for external nutrition immediately after hatching. Also, this study provides evidence that the ontogenetic development of Amphiprion bisinctus is visibly higher compared to other cultured species. So it follows that Amphiprion bisinctus is a species of high importance, and for this reason, further studies should be conducted to contribute to its efficient aquaculture and to prevent the depletion of the local habitat. In conclusion, the study of the ontogeny of the digestive system is very important in order to optimize the larvae rearing conditions and to achieve better production results, including higher survival, better welfare, reduced skeletal deformities, and a low cost. Further studies focusing on the functionality of the digestive system structures, on the digestive enzymes, and other processes which are associated with the feeding acquisition, such as vision, smell, and taste, will complete the necessary information required for the optimization of the larvae rearing procedure. I would like to thank all of the participants from the Hellenic Center for Marine Research and the University of Thessaly. And also I would like to mention that this work is a part of my dissertation submitted in fulfillment of the requirements for the degree of MSc course in Mediterranean aquaculture. Thank you very much for your attention.
Thank you, Mr. Trakaki. Any questions? Yes. Yeah.
Thank you very much for your talk. It was very interesting. My question is just the growth was going in parallel with the ontogeny. Is that right? So the length. You showed a slide that, so it would happen in any case, right? Do you know exactly, so how much more days do you need to reach the final, how much is it delayed from the lower company temperature from these 3 degrees?
Okay, for the specific
Yes, for the species.
Okay, actually, could you rephrase it?
So I realized that the ontogeny is going parallel with the length, right? So the bigger the larvae, the ontogeny, the steps, I mean. So this is actually, the temperature has an effect on the growth, on the length. And then, of course, we see the ontogeny. So how much could we say that is delayed to reach the final stage from these 3 degrees, whether they grow at 23 or at 26? I mean, does it really matter at the end? Is it a big... Or probably this was not the target, but I'm just curious.
Actually, yes, this was not the target.
This was not the target. Okay, that's fine.
Not really matter. I mean that this variation doesn't delay a lot.
Delay, okay.
A lot.
To reach the final stage. Okay. Oh, sorry, no, I'm just asking why would it be important to grow them at 26 degrees?
Why would it be important?
Yes. It's okay. We can discuss it later. No, I said that I realized that at the end they all reach, in both temperature, they reach a final length. So though it takes more time when they grow at 23 degrees. Am I right? Yes. Okay, so I said, what is the delay? Like, let's say if you need 5 days to reach the final length at 26, how many days do you need at 23? Is this a big delay? Is this bad for some reason? Okay. Okay, that was my question. Yeah. If there is a problem when you don't grow them at 26 degrees and you prefer to do it slower.
Because 23 degrees, it's not a very low temperature.
Yeah, okay.
Range. But if the temperature would be lower,
yes, of course.
Maybe possibly a problem.
Okay, great. Thank you very much. Okay. So now Professor Papadopoulos from the University of Thessaly will present his study regarding the effects of melatonin in preserving gilthead seabream spermatozoa at minus 196 Celsius and minus 80 degrees Celsius. Professor Papadopoulos, the floor is yours.
Thank you very much. Good afternoon, ladies and gentlemen. First of all, I would like to thank the organizing committee that giving me the opportunity to present some of our work. And what I want to describe today is an experiment we carried out looking at the effects of melatonin in preserving Zild Hide C prim spermatozoa at minus 196 degrees Celsius and minus 80 degrees Celsius. Why were interesting about cryopreservation? In the industry of aquaculture, the cryopreservation process provides many benefits. Some of them is constant supply of sperm, synchronization of the gamete availability of both sexes, storage of semen for genetic improvement programs that is very important now for a lot of species in aquaculture, reduction in the cost by eliminating the need to maintain male brood stock, transporting the gametes or embryos between farms instead of breeders, marketing the well-characterized and standard quality sperm, and conserving the gametes of endangered species. The process of cryopreservation causes various damages to the level of plasma membrane, on mitochondria, DNA, and RNA. That damages could be used as sperm quality markers. And these markers have an impact. You can use them to see the damages, what the impact they have on sperm motility, fertilization ability, embryonic development, and progeny fitness. Our group in 2022 published a review. The goal of this review was to summarize in the literature the benefits of use of melatonin in cryoprotective solutions, but even for human semen and or farm animals. And what lessons we can take from this knowledge to use for the cryopreservation of fish species. And there are a lot of benefits of use of melatonin. Again, recently we published another review which examines the actions of melatonin via receptors in plasma membrane to ameliorate oxidative stress that is generating from the process of cryopreservation. And melatonin have shown that protects the sperm from oxidative stress, maintain sperm viability, protects from DNA fragmentation, reduce morphological abnormalities in several species, and increases the activities of antioxidant enzymes, as like catalase, glutathione peroxidase, and superoxidase dismutase, that are very important for the oxidative stress. So, we have done an experiment in 2022. The goal of this experiment was to examine 4 different concentrations of melatonin in a cryoprotected solution, 0.51, 1.5, and 2 millimolar in a solution, in cryoprotected solution, and we conclude that there were not some benefits for the quality of semen with these concentrations of melatonin. So therefore, the aim of this study was to examine 2 concentrations, 0.05 and 0.1 millimolar in the cryoprotected solution. If this lower now concentrations of melatonin
could be protective for sperm during in vitro storage at liter Nicogen at minus 196 degrees Celsius in liquid nitrogen and in minus 80 degrees Celsius in ultra low freezer as an alternative method of cryopreservation. Our hypothesis was that the two lower concentrations of melatonin in the cryoprotectant solution could be more protective for sperm during in vitro storage than the higher concentrations that we tested in the previous study that I have shown. In terms of materials and methods, milk of each male with a total motility above more than 80% we used, extended composition with 100 sodium chloride plus 100 BSA. In the control group, we had a cryoprotectant of DMSO at 5%. Melatonin groups, when I mentioned before, 0.05 millimolar and 0.1, two groups of melatonin. The calibration time for cryoprotectants and sperm was during 10 minutes. Preserved sperm samples were thawed at 25 degrees for 12 seconds. Motility parameters were examined, assessment with CASA systems of the company Hamilton Thorne. Sperm samples examined 5 days after a week after freezing and thawing process. And we examined also the kinematic parameters with the CASA system. And the statistics were performed with Minitab software. In terms of experimental design, 4 samples from November to May we carried out in a philosophy company in broodstock facilities at AMPHITHEATER. The 4 samples pooled and we had the dilution rate of solution and sperm 1 to 3, 1 part of semen, 3 part of cryoprotectant solution, and the treatments, the control treatment with sodium chlorate, BSA, and 5% of DMSO, and the 2 groups of melatonin without DMSO in the cryoprotectant solution. This is here we have some pictures when we were assessing the kinematic parameters during cryopreservation and thawing process. And now in the results, if we go to the results, we can see that in the group, in the control group, the spermatozoa was a higher number of mobile spermatozoa than in the 2 other male melatonin groups. And this pattern was the same for even for minus 80 degrees Celsius or minus 96 degrees Celsius. This pattern was the same for the progressive semen, high-speed semen, mid-speed semen, and low-speed semen for in both methods of cryopreservation. In conclusions, our results show that no extra protection was offered by the 2 lower melatonin concentrations supplemented in the cryoprotectant solution. Melatonin as an antioxidant or cryoprotectant in the cryopreservation media may depend on the species. In the case of Spirus aurata, enrich the cryoprotectant medium with lower concentration of melatonin or collection of semen at night. Probably we can have higher plasma levels of endogenous melatonin, could have a better protective effect on spermatozoa protection. And further research is needed definitely to investigate the beneficial exogenous concentration in the cryoprotected media and endogenous plasma levels of melatonin during the cryopreservation of Sparus aurata sperm. I thank you very much for your attention.
Thank you very much for your interesting talk. Does anyone from the audience has any question? Okay, so we can move on. Thank you very much.
Thank you.
And okay, and now let's move on to Dr. Altan, who will present us his study on the effect of earthen ponds to growth, feed performance, and unsaturated fatty acid content of fish meat in rainbow trout on Corinthus micis. Thank you very much. We can start.
Thank you. Dear chairpersons, dear distinguished participants, before my presentation, I would like to express my kind regards and gratitudes from your neighbor, Ege University Faculty of Fisheries, Department of Aquaculture. And as you follow up from the title that today I would like to talk about the rainbow trout and earthen ponds. So first, as you follow up from the title, we will talk about the growth performance of rainbow trout in earthen ponds. And first stage, I would like to explain what motivated us to create this study. The number one is rainbow trout culture is becoming more popular day by day for the Turkish aquaculture industry. Earthen ponds contain an interesting structure with their live food content for the fish, and the age of the fish is quite important for fish release activities from the hatchery to nature. If we check about the popularity reasons for rainbow trout, the fish farmer's perspective, so they prefer to produce rainbow trout very much in my country because of egg and sperm retrieval is quite easy, provides rapid growth and development under optimum conditions and low feed conversion rate, FCR. It's a species resistant to changes in environmental conditions and other stress factors, easily adaptable to mixed commercial feed. A significant part of the disease and breeding problems have been solved because globally there are lots of trout culture experts. Also in my country, we have. There are marketing opportunities in the form of fresh, chilled, frozen, smoked, and trout caviar. And finally, nowadays, the species rainbow trout, also called Turkish salmon, and we just entered to Asian, European, and Far Eastern markets. So it's becoming more and more popular nowadays. Just give an example about the popularity. I would like to make a small comparison in the table. So you can see, according to latest year statistics, we also reached to 200,000 tons per year about the trout production. And as second stage, I wanted to just make a comparison for jilted sea bream and European sea bass that for Greek aquaculture, actually, we are in a sweet competition. 1 year you produce more and 1 year we produce more than you. So this table can give a very good sample about the marine species and rainbow trout culture in Turkey. But on the other hand, I'm not sure about this huge amount is quite good for our aquaculture industry because one side I'm proud of this huge amount, and then my country is making enough money for this. But on the other hand, how logical to spend our freshwater sources to use rainbow trout in, again, freshwater environment. So this is so in this slide, I actually explained most of the information, but in the red line, I just wanted to forward this information according to FAO 2020 and FIAP 2022. Rainbow production in Turkey meets 13% of world production. So according to the data from the Turkish Statistical Institute, Turkey produced almost 200,000 tons freshwater plus marine environments, particularly in Black Sea coast. It's very popular rainbow trout production we have. So in this slide, I would like to discuss about the earthen ponds. In my opinion, it's an interesting semi-intensive aquaculture environment because the first investment and construction costs are very much lower than the intensive systems like net cages or net cages in dam lakes or concrete ponds. There is an organic matter in different types, for instance, live foods, small crustaceae, zooplankton and phytoplankton in the environment. Fish are fed with commercial feed plus live food supports the feeding regime. Live food creates an attractive and brilliant look. This provides a market demand because in Turkey, when you check the market, so if you see the skin of the fish is quite brilliant, so people are going that fish, not to the others. So if we have an opportunity about live food existence in the earthen ponds, so people prefer the fish coming from earthen ponds or marine environment. But on the other hand, we should accept that the fish from the marine environment is always more expensive than the earthen pond environment. But on the other hand, we have some disadvantages in earthen ponds because the stocking density is much lower than the intensive environments. And even in this study in concrete ponds, we stocked the fishes around 15 kilo per cubic meter, but in the earthen ponds, we reduced this up to 7 or 8 kilo per cubic meter. The amount of dissolved oxygen may decrease due to the fertilization in the ponds, particularly in the summer season. And the level of food safety is medium because we cannot control every conditions like we did in the intensive aquaculture. The market price of the fish is higher than in intensive environment. This is maybe this is an advantage, but actually we accept this such an disadvantage. So actually this is very long written slide, so I don't want to go ahead with this, and most of the parts I have already explained. But the most important thing I would like to say that the earthen pond culture become a production model that more and more producers have started to prefer due to lower feed conversion rates and the fact that the consumer thinks that the meat color and taste are very close to the fish in the nature. This is the most important part of this slide. So on the other hand, thanks to the first presenter, the young researcher introduced about the essential fatty acids very much. So I don't want to talk a lot about this. But on the other hand, we should accept that as many of the fishes, the nutritional sources, particularly omega-3 and omega-6 series fatty acids are very important for human life. And here at the second stage, when I was making a literature search, I have also information about this, but on the other hand, came to me a very interesting information. The Salament Eggersdorfer, the researchers, says humankind consumes almost 200,000 tons of omega-3 series fatty acids annually, and almost 99% of this obtained from the fish. This is the most important and attractive part of the motivation of this study also. So I can continue the materials and methods. At the first stage, please accept that this study is one of the first industry and university collaboration studies. This study also was conducted in the private rainbow farm. At the first stage, we provided totally 200,000 rainbow trout were provided from a private hatchery and transported to the private fish farm, to the concrete ponds. And the average live weight is 2 grams. The fish were stocked in 4 concrete ponds, and namely Control, T10, T50, and T100. Actually, we named these type of controls because we wanted to control the fishes when we stocked in the concrete ponds, wanted to transfer to the earthen ponds around 10 grams when they reached to 10 grams, and later on 50 grams, and finally 100 grams. And as control, we kept rest of the fish in the concrete ponds to make a comparison about the productivity or the effects of the growth and essential fatty acids procedure in the concrete ponds and the earthen ponds. So the first transfer group was T10. Actually, the average live weight of the fish around 9.6 grams and started to be fed in the earthen ponds with 3 replicates. And the rest of the groups plus control was kept in the concrete ponds to reach them after to 50 grams and 100 grams to wait for the reaching about these average live weights. And the second transfer group was T50, whose average live weight reached to almost 51 grams. And later on, the last one and the control kept again in the concrete ponds to wait to reach to 100 grams again. And finally, around 150 average live weight, the group T100 was transferred to the earthen ponds, and the rest of the fish were kept in the concrete ponds to see the differences about the growth performance with both earthen ponds and concrete ponds. Here we have the feed and feeding method. We can come to the feeding site. You see the content of the commercial feed. Between 2 grams and 10 grams, we used extruded feed number 1.2. And after 10 grams to 50 grams, we used commercial feed number 2. And from 15 to 100, we used number 3. And then 100 to harvesting period, we used extruded feed number 4, 5, and 6. Actually, it's called commercial names. It means that, for instance, when we called number 2, it's almost around the diameter of the feed is almost 2 centimeters. Fish were fed 5 times a day until they were full or they were satisfied between 10 to 50 grams average live weight, 4 times a day, between 50 to 100 grams, and 3 times a day from 100 to the end of the study. It was 3 times a day we fed the fish. The daily feed amounts were recorded, and the fish live weight developments, feed conversion rate, specific growth rate changes were monitored depending on the weather conditions. Particularly, we followed it in 15 and 30 days. Average live weights of fish were measured with sensitive balance, and aerators, particularly in the earthen ponds, to balance the dissolved oxygen with the concrete ponds, we used regularly the aerators. And here are you see the calculations and the formulations. They are very classical for the fish feeding and nutrition studies. Measurements and analysis, the water temperature, the salt, oxygen, and pH values were measured and recorded daily with OxyGuard to determine the changes in unsaturated fatty acids that can be seen in the fish depending on the growing periods of the fish in concrete ponds and earthen ponds. 10 samples were randomly selected from each experimental group, and they were sent to a private laboratory licensed by the Turkish Ministry of Agriculture and Forestry of Turkey for cold chain storage and analyzed. And for the statistical analysis, we asked the consultancy services to the Faculty of Science in Ege University. And the finalization, we finalized the trial after 196 days. And the final average live weight was 270 grams. So actually this amount was accepted by the fish farm as a marketing size. So before the results about the growth, let's talk about the average temperature. It's around 15 centigrade degree. The average dissolved oxygen 6.8 milligram per liter, and the average pH was 7.75. So according to the results, growth and feed performance of the rainbow, according to the concrete ponds and the earthen ponds, the first one is control, you see, and in red lines, we put the experimental group T10. It was the group that we transferred the fish from concrete ponds to earthen ponds around 10 grams, showed the best growth performance. If you check the initial weight, final weight, daily weight gain, feed conversion rate, specific growth rate, and survival rate, in all matters, T10 was the first group that showed much better results than the others. You see the chart, the number of measurements during the 196 days, and you see the growth development here. And the other important part is the unsaturated fatty acid content of the fish meat. So you see the nutritional value and the fatty acid contents that could be measured by the official laboratory. So actually they couldn't find all the fatty acid diversities here. But when they calculate and measured, so we finally managed to put on this table. So you can see that the T10 groups, the first group was transferred to the earthen ponds, actually showed us very good results. Here, actually, I have a small summary for further studies. Okay, it's the study is not very excellent study, but it was one of, as I mentioned before, it was one of the first cooperation between the private industry and the university. So what can we do for further studies? So if we remind, every earthen ponds have their specific environment, because each farm has another world, another conditions, another environmental conditions, another types of management, another types of fisheries and aquaculture engineering, etc. And also depending on the season, we can measure the diversity of the phytoplankton and zooplankton, because the content of the live food in the earthen ponds may affect using the commercial feeds, the support of commercial feeds. And the live food content should be determined regularly for similar studies. And also we can do some more studies on the stocking densities depending on the intensity in concrete ponds and earthen ponds. So we would like to make an acknowledgement to the Turkish Research Council, because this study was supported by this institution. And here I have the reference list if you want me to share, it will be my pleasure. And it was the final of my presentation. Thank you very much for your interest.
Thank you very much, Dr. Altan. It was a very interesting presentation. It's very nice to see these things happening, very beautiful. So does anyone have any question? Yes, please. Oh. About what temperatures and salinity level we're speaking about for this aquaculture? Because as I know, it's invasive species. And is it difficult to keep this level of salinity and temperature?
So actually, we are talking about the earthen ponds with the fresh water, not the salinity. In my personal opinion, it's such a brackish water, and the total salinity, I don't think that it's much more than 1/1000. That's why I found this type of studies quite dangerous, because even it's such a brackish water, or let's say fresh water, actually, it's another subject of the presentation, because at this stage we can discuss about to use freshwater resources as a carnivorous species like rainbow trout in the freshwater environment, because maybe you followed up according to FAO, Food and Agriculture Organization. In their reports, they say after the year 2050, we are waiting the water wars. That's why, as an expert, okay, I'm very proud about the huge amount of rainbow trout production in my country. But on the other hand, I found this a bit dangerous to use this type of freshwater resources. And the second question, the temperature, the average temperature was around 15 centigrade degree. It's almost good for the rainbow trout, but it accesses to 20 centigrade degree. It's very dangerous for the rainbow trout. So we don't feed the fish in concrete ponds, and both in earthen ponds also. For this study, it was the optimum levels for the temperature.
Okay,
thank you for the question.
Any other question? Okay, so thank you again for this very nice
Thank you very
talk.
much.
Thank you. So let's move on. We have 2 more talks. We will invite Mrs. Papadouli. Okay, she will present us her study she's from the University of Thessaly again, Department of Ichthyology. So she will present us the results of her study regarding the impact of mycobinder products on growth and health of intensively farmed fish. So thank you, Mrs. Papadouli. The floor is yours. You have 10 minutes. Thank you.
Thank you. Good afternoon. I'm Christine. I'm a PhD student at the Agricultural University of Thessaly. My field of specialty is fish health, and especially mycotoxins. So today I'm going to talk to you about the impact of mycobinder products on growth and health of intensively farmed fish. Okay, thank you. So this study was a part of a whole project called Mycotoxins in Aquaculture, Innovative Management Practices to Protect Fish and Consumer Health. There were 3 cooperating agencies in this study: the University of Thessaly, and more specifically the Aquaculture Laboratory from the Department of Ichthyology and Aquatic Environment, the Hellenic Marine Research Center, and more specifically the Laboratory of Pathobiology and Pharmacokinetics, and the Made fish cultures. So this project had 4 main pillars. The first one was to measure the frequency of occurrence of mycotoxins in the aquaculture sector. The second one was to develop an innovative and, most importantly, easy-to-use protocol for their detection and measurement. We wanted to measure the effects of mycotoxins and the efficacy of mycobinders on the health and growth of farmed fish. And last but not least, the application of all these new technologies on a production scale. So why did we bother to do this research? Because of the limited availability of marine protein sources, plant proteins have been widely used in aquafeeds as a substitute for fish meal diets. However, the contamination of plant ingredients with toxicogenic fungi occurs in a wide range more and more regularly on raw materials all over the world. Mycotoxin consumption by fish can result in various toxic actions and raise concern not only for animal health, but also for food safety in general. Mycobinders can be an effective strategy to eliminate mycotoxins and avoid their absorption from the animal. So, mycotoxins, what they are? As we all know, they are secondary metabolites produced by fungi, often found in agricultural products, and diets contaminated with these mycotoxins can be a burden on health and performance of cultured fish. By reducing feed intake, by reducing productivity, by causing greater susceptibility to various diseases, and even in some cases by causing mortalities. So 3 of the most emerging dietary mycotoxins are aflatoxin B1 or AFB1, fumonisin B1 or FB, and deoxynivalenol or DON. So our target was to evaluate the effectiveness of 2 mycobinders of different technologies incorporated in experimental diets against the combination of the mycotoxins that I mentioned to you before, FB, DON, and AFB1, on zootechnical parameters and health indices of gillhead sea bream. Why gillhead sea bream? Because it is one of the most cultured fish in the Mediterranean aquaculture. So we wanted to determine whether the consumption of food contaminated with these mycotoxins could be treated with the use of mycobinders, and if these mycobinders are completely safe to use. So we used 2 mycobinders of different technologies incorporated in the fish diets. The first one, we named it MICBIN1, with the composition of attapulgite plus oxidizing magnesium and activated carbon. And the second one was MICBIN2, with the composition of yeast stain, zeolith, and algae. Those 2 mycobinders were both commercial mycobinders that are used in the animal feed industry. So how we did it? Triplicate aquariums received 3 experimental diets. The MIC diet consisting a cocktail of these 3 mycotoxins that I mentioned to you before, DON, FB, and AFB1, in this concentration that you can see. Diet A1 with the same cocktail of mycotoxins plus the mycobinder 1. The diet B1 with the exact same mycotoxin cocktail plus the MICBIN2. And of course, there was a control group fed with mycotoxin-free diet without any addition of mycobinders. So the fish were fed ad libitum with 2 meals per day for 6 days a week. The feeding trial lasted 12 weeks. Feed intake and mortalities were daily recorded, and the physiometrical parameters of the water were maintained between the standard levels. So about the sampling, We measured the energy utilization by measuring the feed intake and growth, and liver samples were collected for histological analysis to see maybe liver lesions. About the results, the MIC group had tended to have the lowest feed consumption, and the control group had the highest one, as expected. About the body weight, again, the mycotoxin group, the group that was fed with only with mycotoxins, had the lowest body weight, following by the other three groups. Same happened to the weight gain. The control group had the highest weight gain, and the mycotoxin group had the lowest weight gain. The groups with the addition of both groups with the addition of both mycobinders had better results than that of the mycotoxin group. Same happened for the FCR. The mycotoxin group had the highest one. Now, about the histopathological results. Here you can see a photo of the control group with no addition of mycotoxins or mycobinders. You can see a completely normal hepatic parenchyma and hepatocytes. Now, the mycotoxin group, as you can see, showed severe diffuse degeneration of the liver parenchyma with vacuation of the hepatocytes, some areas even showing necrosis and loss of normal hepatic liver structure. Now, about the MICPEN1 group, we showed mild degeneration in small areas of the liver with vacuation of the hepatocytes, but also there were areas that were completely normal. And about the MICPEN2 group, there was mild diffuse degeneration this time of the liver with vacuation of the hepatocytes. To conclude, the dietary addition of mycotoxin cocktail showed negative effect on growth and health of gillhead sea bream. And we saw that the nutritional addition of mycobinders, of both mycobinders, appeared to have a positive effect on shrinking the problem, but not eliminating it. Thank you very much for your attention.
Thank you, Ms. Papadouli. Any questions? Yes, please.
No, no, not something considerable that was caused by the mycotoxins or the mycobinders.
What do you mean considerable?
I mean in the 30-25 phase of the of the exact aquarium of the aquarium, we showed 1 or maybe 2 deaths.
Another question? Yes, please.
At libidum, until the fish is full. Until the first pellet stays on the, not on the surface, down to the aquarium, on the bottom.
Okay, thank you very much. And the last presentation from Mr. Soikan, which one is a safer occupation, fisheries or aquaculture, the case of Turkey.
Thank you, Chair. Ladies and gentlemen, this is my second presentation for today. And in my first presentation, I already mentioned, but I would like to express my thanks to the organizing committee of
HydroMediT 2024. So thanks for organizing such kind of an event. So today I will try to explain you about the occupational health and safety of Turkish fishing and aquaculture industry. Maybe when you see the title, you already have an opinion, but let's see if you are on the right way or not. So let me start. But before start, when the topic is about occupational health and safety of fishing and aquaculture industry, we can say that the authorities generally close their eyes. So that's why I wanted to make a speech on this subject to increase our awareness on occupational health and safety, not only in Turkey, but also in other parts of Mediterranean and in the world. So let me start. Occupational health and safety, briefly it's OHS, in the field of fisheries and aquaculture involves special challenges arising from the nature of the sector. Workers in the aquaculture industry often operate under hazardous conditions, which bring various occupational health and safety risks. Fishing and aquaculture workers face a range of hazards and risks on the job, including, but are not limited to, such as drowning, falls overboard, entanglement in fishing gear, exposure to hazardous weather conditions, and musculoskeletal injuries from repetitive motions. Now we go to some statistics. We know that number of fishing vessels around the world is more than 4 million, and almost 60 million people are engaged in fisheries and aquaculture. And according to FAO, the fatality rate for fisheries is 100 over 100,000. And these statistics make fisheries and aquaculture industry a very dangerous job. Fishing workers are responsible for harvesting fish and other marine life from the ocean or freshwater bodies. They work in a variety of settings, including commercial vessels, fish farms, and production plants. This is a statistics from the United States, and you can see that regarding the fatality rates, fishing is in the top 10 occupations. And we have statistics from the European Union, and also we see that almost 20% of all fatalities in all occupations, unfortunately, based on agriculture, forestry, and fishing. So when we talk about the case of Turkey, a limited number of cooperative studies indicate that Turkish fisheries and aquaculture industries have one of the highest incidence rates for fatalities and permanent incapacity, together with mining, construction, and transportation. This is the general overview of Turkish fishing industry. You can compare what we have in fisheries on the left and what we have in the aquaculture industry. Despite the fact that fishers and aquaculturists have one of the most dangerous professions in Turkey, only limited research efforts have been made so far towards improving occupational health and safety in this industry. It's also noteworthy that studies regarding the statistics in-depth analysis of occupational incidents in the Turkish fisheries and aquaculture industry, such as fatalities and major injuries, are missing. The aim of the study is to compare health and safety data between capture, fisheries, and aquaculture on some parameters of occupational health and safety. Obtaining results may serve as a valuable source for occupational health and safety management of each subsector. So when we come to the materials and methods, the data of occupational accidents, in some references, it's also referred to as occupational injuries. So it's the same, occupational injury and incident or accident. And the precise number of employment in the fisheries and aquaculture industries from 2006 to 2022, which is a 15 years data set, presented in this article are collected from yearbooks of work accidents and occupational diseases statistics and insured workplace statistics of the Social Security Institution of the Turkish Republic. So this study is based on official data of the authority. These documents are the only sources reporting occupational accidents and employment in Turkey. According to the Communique on Occupational Health and Safety Danger Classes of Economic Activities, the Turkish fisheries and aquaculture industries is divided into 4 subcategories: marine fishery, freshwater fishery, marine aquaculture, and inland aquaculture. All occupational injuries are subject to being reported to the authority, regardless of how serious the injuries are, within 3 working days of the incident has happened, according to the Turkish legislation. In this study, 3 descriptive occupational health and safety parameters, incident rate, permanent incapacity rate, and fatality rate, were used to compare fisheries and aquaculture industries. And actually, the main reason why I use these 3 parameters are the only available parameters in the statistics. The milestone for Turkish industry in terms of health and safety was the enactment of Occupational Health and Safety Law, which was put in force in 2012. Related to the law, fisheries and aquaculture are in the category of dangerous occupations in the Communique of Safety Danger Classes. There are 3 danger classes: very dangerous occupations, dangerous occupations, and least dangerous occupations. And fishing and aquaculture industry is located in the middle. So these are the subcategories of fisheries and aquaculture industry in Turkey. So let's talk about results. Here you can see a comparison between Turkey in general and what we have in fisheries and aquaculture industry. I know that tables are a little bit hard to understand, but when we go to the figure, you will be more clear that when you look at the fatality rates, in some years, we can see that fatality rates in fisheries and aquaculture is over than the Turkey in general. And I would like to support this finding with some graphics. You see, in some years, the blue line is the general mean of Turkey, or Turkey in general, and the orange line shows the fatality or incident rates or permanent incapacity rates of the industry, fisheries and aquaculture industry. And regarding the fatality rates, you can see that in some years, it's over the average of Turkey in general. This is also a comparison between fisheries and aquaculture regarding it includes employment, incidents, permanent incapacities, and fatalities. And at the end, we can see that 10 years data set show that we have totally 12 fatalities in the aquaculture industry, while there are only 9 fatalities in the fisheries sector. And this is also another descriptive statistic image. You can see the fatality rates, incident rates, and the incident trend of the occupations based on each subsector. And for example, if you look at 2020, we see that number of fatalities in marine fishery is 4. And for example, when we go to the marine aquaculture in the same year, there are only 2 fatalities. So if we compare fisheries and aquaculture with some trends, we see that the trends in aquaculture are also reasonable, more reasonable than fisheries. When you look at the production and accidents in the fisheries, we cannot see a similar trend. But when we go to aquaculture, the trend is more or less similar. It's also very, very clear when we go to relation between employment and accidents. And if we compare fisheries and aquaculture with the status of Turkey in general, we can also see that the statistics or the data in aquaculture is more relevant with the status of Turkey in general. Regarding the fisheries, there are fluctuations, especially in accidents and employment. Actually, this topic, as I started in my speech, is generally less studied area of science of the fishing and aquaculture industry. But also, we can compare data from other countries. For example, regarding the fisheries, if we compare, for example, Norway, Iceland, Denmark, England, Poland, Canada, United States, and Turkey, we can see that the minimum ratios regarding fatality rates are found in Turkey. Also, when we compare aquaculture, for example, with Norway, we can see that the fatality rates is 1/10 of the ratio in Norway. As I mentioned before, the Turkish statistics regarding the occupational health and safety in the fisheries are lacking too many parameters. For example, mode of injury and injury types, body parts injured in occupational accidents, unfortunately, are missing in Turkish fisheries and aquaculture industry, which are very, very crucial for the improvement of occupational health and safety management. So what to do or what to improve are the trends of occupational injuries in Turkish fisheries and aquaculture. We need more accurate data. And also the quality of data and reporting in Turkish fisheries and aquaculture industry is a must to be developed. Underreporting of occupational injuries, I think this is a very common case all over the world because underreporting seems the biggest barrier to improve our awareness on occupational health and safety. Also, influence of the legislation is a very important area because when you put a legislation on occupational health and safety, please be aware that it may have some negative effects as well. Especially, we can link this to the underreporting case. And the biggest gap of the system, status and awareness of occupational diseases in the Turkish fisheries and aquaculture. During the 15 years data set, no occupational illness has been reported so far. So this is the problem of the data. Some suggestions. We need to improve our education and awareness on occupational health and safety issues, especially by onboard educations, onboard trials, especially regarding the first aid and emergency situations. We need to improve and widen our knowledge on precaution and prevention activities. And also, we need to perform, or we need to create a very, very high control mechanism for the aquaculture and fishing industry. And this is a very common PUCO cycle. So, highlights of the study to conclude. Aquaculture in the 10 years data set, totaling with 12 deaths, fatalities, unfortunately. Fatality rate of aquaculture is 5 times greater than that of fisheries. And cumulative number of occupational accidents and fatalities in aquaculture was greater than that of fisheries, although the number of employees is less, according to the statistics. Trends of aquaculture based on occupational accidents are parallel with the general work life of Turkey. And we can say that the Oscar goes to aquaculture for this presentation. So these are the references. And thank you for your attendance, for listening. And maybe we can collaborate for further international studies. Thank you.
Thank you very much, Soikan, for your very interesting presentation.
Thank you.
Any questions from the audience? Yes, please.
Can I take the question again, please? Well, Turkey is a whole. So I cannot give you a very strict answer about it. But when you consider about the Turkey's geographical position, all aquaculture facilities are located, or most of them are located, on the Aegean coasts of Turkey. So most of the data comes from that area. We have also inland facilities, inland fish farms, and inland capture. But the data from inland fishery is very, very less than the marine fishery and marine aquaculture.
Other questions? Yes, please.
Thank you, sir. Yeah. Occupation, yeah. Yeah. Definitely. Incidents, yeah. Yeah. Yeah, thank you. Yeah, thank you for contribution, and that's why I would like to collaborate with colleagues. Thank you.
Okay, very interesting dialogue, and the session has finished, and we are very happy that joined us in this session, and now there is a break. Thank you very much. Yes. Yeah. Yes. Okay.
Thank you.
This Γεια σας.
Γεια σας. Ωραία, σας ακούω μια χαρά.
Ωραία. Εγώ θα κάνω share, έτσι.
Εσείς, ναι.
Να κάνω μια δοκιμή.
Όχι, δεν χρειάζεται, γιατί ρίχνω τώρα το banner. Δεν χρειάζεται, απλά θα κάνετε όλη την οθόνη όταν θα έρθει η ώρα.
Εντάξει, αν δω ότι δεν μπερδεύομαι μερικές φορές, θα μου πείτε.
Οκ, ναι, αν υπάρχει κάποιο πρόβλημα θα σας το πούμε για να το κλείσετε και να το ξανανοίξετε και να κάνετε όλη την οθόνη.
Εντάξει.
Ευχαριστώ.
Κι εγώ.
Good afternoon. Ladies and gentlemen. Good afternoon, ladies and gentlemen. We will start the session Economics and Marketing of Fisheries Products in the field Blue Economy and Governance. The first presentation, the title of the first presentation is it acceptable to use fishery discard in pet foods? We invite Mr. Aydin, Department of Fishing Technology at the university. Thank you.
Welcome, everybody. Today, I want to introduce you our previous Erasmus project, the details of the project, which includes two important topics. First one is discard fishery, and the other one is pet food. And we just explain what we did in the project, how did we start the project, and these kind of things. I'll firstly, starting with introduction, sustainable food management is necessary to benefit more efficiently from the share allocated to human consumption due to the increasing world population and decreasing the food resources. And in the EU, the estimated percentage of households owning at least one pet is 46% of all households. And there are 77 million dogs and 83.5 million cats in the EU. According to FEDAF, there is annual sales revenue of 50 billion euros in 2021 for pet food and related services. The sales value of pet food products in 2021 was 27.7 billion euros. And the amount of food produced for feeding of these animals has reached 8.5 million tons per year, which is estimated to be more than $100 billion in monetary value. So, pet food industry is growing 2.6%. That's why the need of raw materials of animal origin is increasing day by day. And this is the other path. About fishery, all of the dead or living animal origin material that does not contain plant products and residues that is thrown back into the sea for any reason in the total catch is called discard. And what is discarded fish? And I just want to ask a question to everyone. Think about that. You're hungry and you have money in your pocket. And this is one of the most famous fish of Mediterranean, strip red mullet, Mulus rumletus. And think about that. You like to eat fish. If you have money in your pocket, do you want to buy this fish to eat? Please give answers. It's legal size, caught in the legal area. Do you want to buy it? No. Why? It's not small. It's legal size. Think about this. It's not small. It's legal size. Everything is legal. And do you want to buy it? Probably everybody wants to buy it who wants to eat fish. However, do you buy this? No. Why? Because of damage. Yeah. Nobody wants to buy this fish individual from market, but it's commercial species. However, the most important reason for discard fishery is discarded commercials. And the reason of them is changing about holes, about some scavengers, also some of the sea turtles, also some of the reasons coming from fishing gears. Sometimes some commercial species can easily discard it for this reason. And also, there are lots of reasons discarded in fishery. The most important one maybe is the type of the fishing gear. You can see the sardines in here, and sometimes fishermen easily catch some sardines in bottom trawl fishery. However, it's not huge amounts to sell, but it's commercial if you look. Then they can, they don't want to sell because it's not high commercial as the other, like the other bottom trawl species. I mean, red mullet and hake and the other ones. This is the one of the reason type of the fishing gear. Also, feeding habits of related public. I think this is also a very important reason between us. We are sharing the same waters, adjacent waters we have, Greece and Turkey. And these photographs were taken from your fish markets. And they are commercial. You can eat this fish. Also, we can eat this fish too. However, our public, our people, don't want to eat serranid species. We are really very luxury in eating fish. We just know, we just want to eat, my public just want to eat some 5 or 6 kind of fishes. I mean, sea breams, sea basses, mullets, hake, and in the wintertime, anchovy and sardines like this. And this is one of the reasons of discarded species from bottom trawl in Turkey. And also selection of the individuals after harvesting is also another reason. This reason comes from the crew of the fishing vessel. Sometimes crews don't want to select lots of fishes from the hole, because sometimes they have some cold, or they are very sleepy, or the weather conditions are very bad, and they just want to finish the work and go inside the fishing vessel. That's why you can easily find these kind of commercial species in the discarded amounts. So today we are seeing 30% of the world's fish stocks are overexploited. FAO estimates that total fish production is 179 million tons in 2018. And about 88% of this amount was utilized for human consumption directly. The rest was described as for non-food purposes and waste. And how about selective studies? Yes, we did. Bycatch reduction, sorting grease, T90s, square meshes. Yes, we did lots of research projects, scientific projects, all of the world. Yes, we were success. We did something. We reduced the discard. However, it's not practically usable for commercial fishermen. Fishermen, yes, understand what we did, but don't want to use it because it's not practical for them. That's why it's really impossible to reduce the discard in the fishery. It's not impossible, but it's too hard to reduce and finish the discard in the fishery. This is just the reason is because of the fishing habits of the fishermen. And also in Turkey, we know that 430,000 tons of demersal marine fish were captured. And also the calculated estimation is approximately 45% of them were thrown back to the sea, discarded. You can see the discarded amounts, discarded photographs. Also, the other step. Cats and dogs are carnivores. Also, dogs are omnivores. And they have same differences in their nutrients need and nutrient digestion. So, this is the 3 types of the pet foods in the world. The bottom, you know, is dry food and cooked and have lots of plant-based products inside. Top, canned food, yes, maybe more healthy, but also it's cooked. But you can see the red one, like a mince, it's barf. Okay, barf. Yes, it means biologically appropriate raw food. And we can easily put ingredients of barf from fish products, fishery products. That's why we created a project, an Erasmus project, includes 5 sustainable development goals of United Nations. It calls MARIPET, and this is the website of our e-learning platform. You can easily use this address and also our Instagram account. And you can see on the right side, our partners coming from Norway, Iceland, Lithuania, and Croatia. I should be quick because the time is come. And also, wasting food feeds climate change. This is an important reason. And I also lost my dog, and my dog Alf was the starting point of this project, and that's why I want to put his photograph on this presentation. And thank you for attendance.
Thank you very much.
I should be quick. That's why.
Do you have any question? I have one question.
Yeah.
Do you think that there will be the crucial mass for doing?
Excuse me again.
Do you think that the quantity that you will have will be enough for processing?
You mean the fishery products?
Yes.
Create barf. Yes.
Okay.
Yes. We believe this. That's why we applied the project and we finished it.
Thank you.
Question?
Sorry.
Excuse me. Yes, for EU legislations, yes, this is obligated for EU countries. Yes. Maybe the solution could be giving some money for them for discard. They can put the discarded fish into a basket with ice and you can go and someone go and take all of the discarded fish. If, look, if fisherman believes that they can earn some money, they can do everything. I think the solution is this. Thank you.
Okay.
Thank you.
The next presentation is the. Thank you. The next presentation, the title of the next presentation is Applying Fair Principles to Micro CD Data, the case of Mapworms project. Mrs. Mavraki will present it. Mrs. Mavraki?
Yes, hello. You can hear me, right? I cannot hear you.
Yes, we're here.
Okay. I'll try to share my screen. Let me know if it is okay for you. If it is okay, I can start.
Yes, we're here. It's
okay. Thank you, because we cannot hear very well online. My apologies for not being able today to be there with you. My name is Dimitra Mavraki. I'm a data manager in the Hellenic Center for Marine Research. And my presentation today is applying FAIR principles to micro-CT data in the framework of Mapworms project. I will start with the FAIR data concept that was introduced in 2016 by publishing in the scientific data journal the FAIR guiding principles. FAIR is not a standard, but it's rather a set of principles and standards for findable, accessible, interoperable, and reusable. Both FAIR and open data can make clear the importance of sharing data, information, and access, allowing collaboration and saving cost and time between scientists and users. As I suppose you already know, in Horizon Europe projects, European Commission requires from beneficiaries to manage the digital research data generated in the action responsible in line with the FAIR principles. And that means we have to establish and regularly update the data management plan. In this data management plan, we have to follow the principles, the FAIR principles, and deposit the data in trusted repositories. This is a case for Mapworms project that I will give a short introduction. Mapworms is a Horizon Europe research project that was started in 2022, and it is focused on the development of bio-inspired shape morphing robots. Its main aim is to design robots that are inspired by simplified forms of marine worms, marine annelida. In this project, 6 partners were participating, representing research institutes and universities, and also SMEs from 5 different countries. We have already published the first draft of FAIR data management plan for this project. The Mapworms data management plan aims to define the management steps and protocols in order to inform and guide the researchers involved in the project in properly managing their data and software. We follow the Horizon 2020 FAIR data management plan template, and it's crucial to underline that it is considered as a living document which will be revised following the project progress and mature based on the progress achieved through the project's life cycle. Mapworms project is a multidisciplinary project, and this is important for us to have a FAIR data management plan in place. As I said before, Mapworms, it is a multidisciplinary project, but I will concentrate here, I will focus on micro-CT data. In the Mapworms project, the microcomputed tomography, micro-CT, it's a non-destructive imaging technique that has been used to visualize the morphological and anatomical characteristics of marine annelida. The creation of 3D representations of samples, as you can see here in the short video, as well as the ability for 3D analysis, make this technique a valuable tool for several biomedical and biological research fields. Why? Because micro-CT scans can be used for the calculation of basic morphological and anatomical parameters, which are essential for the shape morphing modeling, in our case, of the robot. However, we have some constraints because there's no metadata standards. There's no large depository for this or data organization. And we'll try to follow a verification process in order to reply to these problematic issues. So I will start with the verification process with the findability principle, the letter F, which means that both metadata data should be easy to find for both humans and computers. This is the reason why we applied unique URL for each dataset that we upload in our repository in order to be distinguished and every user to be able to find it easily. The second principle is accessibility. Data become more accessible if they are published in an open format and if it is clear for users which kind of software is required to read the dataset. This is the reason why we are using the open source software tool, the SliceDrop, which enables the users to download and view the scans, the videos, and also the photographs. The third principle is interoperability. Data must share a common structure and metadata must use recognized descriptions. For these reasons, as you can see in this screenshot from our micro-CT lab, we provide an explanation for every single field of metadata and data records in order to get the user familiar with every field. We also use search filters. For example, the user can select the project and find the appropriate data sets that he's willing to find. The last principle is reusability. The ultimate goal of all is to optimize the reuse of data. And here again, you can see in the screenshot that we provide an extensive list of metadata records. And also at the bottom, you can see that all the datasets, images, and videos are available under a specific license. Here, I summarize the verification process for micro-CT data. You can see the targets achieved for every single principle, but I will focus on the future targets. Currently, we do not apply DOIs. That means that the micro-CT VLab does not assign digital object identifiers, only URLs, but we will do it in the future. Regarding interoperability, we would like to incorporate control vocabularies when defining both data and metadata parameters in the future. For example, to use the Darwin core standards. And I will finalize my presentation regarding the verification process for the whole project, for the MapWorms project. I would like to emphasize that partners are encouraged to be generous and extensive when providing metadata information, and also metadata are accessible even when the data are no longer available. To stimulate discovery, potential reuse of the project's data, we have created a MapWorms Zenodo community, which is accessible through this link, so we can learn more things about this project if you are interested in. And finally, metadata data from the sampling campaigns of the project will be submitted and published in the Mediterranean node of OBIS, MEDOBIS, while all microCT data will be stored and published through the microCTV lab. And I would like to say here that both Medobis and microCTV lab are hosted in the Hellenic Center for Marine Research based in Crete. Thank you so much for your attention.
Any question about this presentation? Okay, thank you very much.
Thank you so much.
The presentation was very interesting, Mr. Mavraki. Thank you very much. Now we go to the next presentation. Extraction of bioactive compounds from plant byproducts intended for fish feed supplements. Laboratory of Process Analysis and Design, School of Chemical Engineering, National Technical University of Athens. Mr. Steriopoulos.
Okay, thank you very much for the small introduction, and good afternoon, everyone, and welcome to my presentation entitled Extraction of Bioactive Compounds from Plant Byproducts Intended for Fish Feed Supplements. So starting with a small introduction first, it is very well known to all of us that the global population is constantly increasing, and it's expected to grow to 9 billion people by 2050. So this causes an even increasing demand for food supply, and aquaculture will play a major role in these dietary demands. Actually, aquaculture represents the fastest growing food production sector, and it has recorded an annual growth rate of 6.5% over the past 3 decades, and it's forecasted to increase a further 15% by 2030. So this rapid growth introduces risks and hazards, not only to the aquatic ecosystem, but also to social equity. So there is a requirement for new, sustainable, and equitable aquaculture development strategies. According to the 2013 Blue Growth and the 2021 Blue Transformation Initiative of the Food and Agriculture Organization of the United Nations, there is a demand to minimize environmental degradation, biodiversity loss, and unsustainable use of resources while maximizing economic and social benefits at the same time. In that aspect, the extensive use of additives such as antiparasitics, antibiotics, and growth agents in fish feed have proven to be costly and environmentally unsustainable. So here comes our project, the Feed Active project, which is a Horizon 2021 Marie Skłodowska-Curie action, a joint research and innovation project with strong partnership involving 8 partners from 3 European countries, namely Romania, Greece, and Italy. The Feed Active project focuses on the development of innovative aquaculture feed supplements based on natural bioactive compounds that boost the immune system and exhibit antioxidant and antimicrobial activity. These compounds are also known as phytobiotics, plant-based natural compounds added into diets, resulted in improved animal productivity and immunostimulation. So here you can see the objective of the work, which is the extraction of these bioactive compounds from plant byproducts that can be used as feed supplements. So the work, you can see also the work overview. First, we selected the plant byproducts We extracted the bioactive compounds using green extraction techniques. We evaluated the extract antioxidant capacity and phenolic content, while at the same time we determined the extraction yield. And in the end, we identified which raw material leads to the richest extracts. So the plant byproducts that we selected are the lemon peels from the genus of Citrus limon, the orange peels from the genus of Citrus sinensis, a mix of these two peels, and olive leaves of the tree Olea europea, and more specifically from the Greek cultivar of Kolovi, which is an abundant cultivar on this very island of Lesbos. All raw materials were provided by local Romanian and Greek producers. Prior to the extraction, we dried overnight the raw materials into a vacuum drying oven, and we further homogenized them by grinding. The green extraction technique we used is the ultrasonic microwave assisted extraction, and the parameters of the protocol involve a temperature, an ambient temperature, a time of 15 minutes, and a constant solid to solvent ratio 1:20, which was optimized from previous experiments that we have conducted in our lab. At the same time, we investigated the effect of different solvents on the extraction of the bioactives, and namely, we used water, a mix of ethanol water with 80% ethanol in the composition, and ethyl acetate. For the determination of the extraction yield, which means how much weight of dry extract we get from the raw material that we use for the extraction, we vaporized the whole extracts, and we calculated the extraction yield as a percentage. And here in this table, you can see the results for all the raw materials. And as you can see, the mixture of ethanol water resulting into the highest yield values for all the raw materials that we investigated. And here you can see also a graphical abstract to show that it's becoming more clear to you. So the orange peels actually resulted in the greatest yield, followed by the mixed peels, the lemon peels, and last one, the olive leaves. For the determination of the antioxidant capacity and the phenolic content of the extracts, for the antioxidant capacity, we determine it by the Trolox equivalent antioxidant capacity assay, which actually is the capacity is expressed as milligrams of Trolox equivalents. Imagine Trolox as a superhero antioxidant on which you in some way express the compounds that show this antioxidant activity as milligrams of Trolox. And for the total phenolic content, we use the Folin Kyokalteu assay, and we express the phenolics as milligrams of gallic acid equivalents. These two protocols as phasmatophotometric, as you can see from the picture, so we use a phasmatophotometer for that cause. Here you can see the results, and again, the use of ethanol water as an extraction solvent for all materials resulted into the highest total phenolic content and antioxidant capacity for all the raw materials that we investigated. Here you can see also a graphical abstract, and I think it's very clear that the most potent raw material proved to be the olive leaves. Here you can see a principal component analysis, score plot, and loadings plot of the parameters of the different assays that we conducted. You can see from this plot from the two first components that actually Trolox equivalents and total phenolic acids actually correlate with each other. And the closest to them is the SA2, with SA is the olive leaves extract with the use of ethanol water as an extraction solvent. Closer to the extraction yield is the RAN5. RAN5 is the orange peel extract with ethanol water 80-20, and although it didn't show a great total phenolic content and antioxidant capacity, the greatest yield that it exhibited can be attributed to a great sugar content that the orange peels have. But what is the difference between these extracts? What is this property that makes the olive leaves extract more potent than the other ones? Here you can see chromatographic analysis. The difference is actually that phenolic acids and flavonoids exist in both extracts. However, the olive leaves contain more antioxidant categories of compounds such as tyrosol and hydroxytyrosol derivatives, oleuropein, and carotenoids, which exist also, for example, in the lemon peel, but in scarce amounts. So to sum it up, we can say that the use of ethanol water proved to be the best extraction solvent in all cases of raw materials. The use of ethanol water resulted in higher yields and extracts richer in antioxidants and phenolics in all cases of raw materials. Olive leaves resulted into the best, into the greatest phenolic content and antioxidant capacity. And this can be attributed to the fact that the olive leaves contains more antioxidant groups such as tyrosol, hydroxytyrosol derivatives, oleuropein, and carotenoids, and the valorization of olive leaves through extracts implemented in fish feed could benefit fish aquacultures by replacing synthetic compounds, decreasing environmental pollution, contributing to health and well-being of fish, and leading to a sustainable way of fish feed management. Of course, the extracts need to be analyzed in respect of their antimicrobial activity, and before implementation, they need to undergo toxicological tests. These tests are actually gonna be conducted by our beloved colleagues here in the University of the Aegean. The addition of fish feed will be into free and encapsulated form. The benefits will be assessed through fish feeding into experimental fish stacks in this very university. Investigation of other plant extracts and essential oils are also being considered to be used. And of course, we will follow with a sustainability study through the life cycle analysis. So I reached the end of my presentation here. Thank you very much. If you need to find more information about the Feed Active project, you can visit our official website and also our official accounts in our social media. Thank you very much.
Thank you very much for your presentation. The results were very interesting.
Yeah, I think so too. And I think that, you know, the aim of the project is to valorize byproducts. So a raw material that we actually throw away, trash. And since the best raw material was the olive leaf, and you know that Greece is full of olive leaves, and of course, this very island is like, I don't know, it's like there are olive leaves to the sea. You can actually, this is, imagine what a treasure we have and how we could use this treasure to our benefit and a sustainable way of.
All this in the discussion, you had several points. Are all these phases included in this project, or you plan a second?
They are all included, and they are already scheduled to be contacted. And here also we have our colleague from the University of the Aegean, Dr. Sofia Papou, who could add more things to the experiments.
Yes, Sofia.
Yeah, we have produced feed and we have implemented already the extracts.
Congrats. Is there any other question? Yes, please. The microphone, please. Yes, but where is the microphone?
Mm-hmm.
Could you please repeat the question because now the microphone is okay?
Thank you. Congratulations for this very interesting work. It was expected to find higher concentrations of oleuropein and all the other antioxidant substances in the leaves of the olive trees. It was expected in comparison to the other plants. But I wonder, the extraction that you have in ethanol, how it is used as a feed additive to feed the fishes, so the ethanol is not toxic. So the concentrate that you have at the end, that you want to use it further.
Thank you for your question. The implementation of the extracts is in dry form, so we totally evaporate all the solvent. There's nothing left behind. Even if there is some small, tiny amount of ethanol, it's the implementation to the feed is like less than 1%. So it wouldn't affect in a toxic way the fish. Also, all these extracts have undergone toxicological tests anyway. So we don't just put it in the feed. We analyze first the results from the tests. We totally evaporate the solvent, and then we implement it in small amounts. It's not like 50% of the feed is the additive. It's like 0.01% in the total mass.
So what you extract there, it's only polyphenols or also other substances?
In the olive, you mean? In the olive.
The olive trees, because I think from there you have finalized the work.
Yes, there are polyphenolics, carotenoids, and there is an amount of chlorophyll as well. These are the basic compounds. Now, yeah, these are the basic compounds actually, from what I'm trying to contemplate now and to remember. But I think, yeah, these are the, so it's polyphenolics, carotenoids, and chlorophylls are the three main group categories, let's say. Thank you.
Thank you very much.
Now Mrs. Argyroux from NOW's consultants of environmental planning and project development will present us the work titled Visualizing the Future of Aquaculture in the Greek Seas, a foresight exercise. Mrs. Argyroux.
Thank you very much. Good afternoon. My name is Ioanna Argyroux. I am a biologist ichthyologist, and I have been working in fisheries and aquaculture sector for 30 years. The title of the present work is Visualizing the Future of Aquaculture in Greek Seas, a foresight exercise. The work that I will present to you has an emphasis on the methodological part. It is a foresight exercise and tries to outline the future of aquaculture in our country. The aim of the work is to feature potential future developments of the aquaculture sector in the Greek seas. The sector is evolving in an environment with significant challenges that is rapidly changing, and the effort is to take advantage of the foresight approach in order to see what the future options of the sector in the Greek area can be. Aquaculture sector is of great interest for a number of reasons shown on the left part of the slide. A branch of the sustainable blue economy, a dynamic sector, a sector significantly contributing to fishery product market demand, a sector offering qualitative and hygienically safe products, a pillar for local development. The definition of foresight is one has been formulated since 2010. Essentially, it is a way to investigate future developments, to gain deep knowledge of what can happen and how this can shape, among other things, the sector we are interested in. Therefore, in an environment characterized by complexity and uncertainty, the tools of foresight can shape a triangle, which is presented here. Debating the future, thinking about the future, shaping about the future. We stand at debating the future, trying to formulate possible future situations. The methodological framework comes from planning and enables us to use foresight tools such as the scenarios. In the context of this work, we proposed two scenarios. We also created a narrative. In conclusion, foresight is a strategic approach to explore the future and map out a strategy within different future images. We apply this exercise in the Greek seas on the specific sector aquaculture and especially marine aquaculture, with the aim of delving into the sector. Marine aquaculture is very important for the Greek economy. We have been working on the specific sector aiming at sustainable, competitive, spatially balanced marine aquaculture with reduced conflicts and creation of synergies with other sectors. Regarding the methodological approach, first we investigate the external environment, the key drivers of change, so as to be able to see through the various developments. The key drivers are understood as important forces that change reality. We also explore the internal environment, current state of aquaculture in Greece, regional considerations, current spatial and legislative context. Based on this information of the external and internal environment, the foresight exercise is taking place, and we end up the prevailing scenario of aquaculture sector. On this slide, we can see the key driving forces of the external environment in a global level and in a national level. Concerning the global level, we mark climate change, market-based trends, dietary patterns. Concerning European level, we mark common fisheries policy, the Green Deal also. Concerning the internal environment, we elaborated a study of the aquaculture sector in Greece, a market-based analysis, strategic guidelines for the spatial development of aquaculture, and spatial development and smart specialization. In a third step, we structure and access alternative aquaculture scenarios. In the next slides, we can see step by step the issues we examine in the external and the internal environments. In the external environment, we mainly examine a wide range of policies that indirectly or directly approach the sector we are studying, is developing strongly, and is related to maritime strategy, coastal zone, habitats, and at the top of them is the Green Deal. Accordingly, we examine policies that have to do with the aquaculture sector itself. Also, the regulation that is referred to the common fisheries policy. A key point of the external decision environment is dietary patterns, intensifying efforts to meet global demand of food by 2050 are increasing. Due to EU fisheries restrictions, aquaculture can be a long-term solution. And correspondingly, technology today offers many opportunities for offshore farming. This is a new possibility that can be exploited and is of interest in the case of Greece. Technology in this field has been intensified. Research and innovation are very important. Besides, many problems are solved in the open sea, making an environmental decrease of the impacts of aquaculture or in synergies between different sectors that can coexist with aquaculture, for example, renewable energy sources. It can be seen here, the two figures from 1986 to 2018, the share of the different branches of internal waters and marine waters from which it appears that marine aquaculture prevails in terms of production compared to aquaculture from inland waters. So here in this slide, we look in depth at the specific sector. What are its characteristics, what kind of businesses, and others. We see here the spatial distribution of aquaculture, and this is a map coming from the Special Framework for Spatial Planning and Sustainable Aquaculture Development, which was issued in 2011 and which is under revision. This is a picture. Areas here in the coastal area or in the archipelago, which are shown as areas for exercising the activity here. We are also interested in the spatial part. The present work tries to emphasize toward this direction. Aquaculture sector is also very important, as other speakers said, and mainly maintains social and economic cohesion and ensures nutritional adequacy. We also explore a policy framework of the internal environment, national strategies, regional strategies, Law 4269 of 2014 for areas of organized development of aquaculture activities. And this is the concept of the foresight exercise. Through the research that has been carried out, the key drivers have been identified and are listed, or better, included in this figure. The two uncertainty axis model is used as key drivers of change. We chose the technology, which can rapidly change the image of the future. This model produces 4 distinct future scenarios. With this approach, the 4 scenarios are built. One axis is technology, you can see here, and the other axis is spatial dimension. So we have high technology penetration or low or medium technology penetration. We have also concentrated pattern of aquaculture development and the concentrated pattern of aquaculture development. From a preliminary qualitative assessment, we have a rejection of scenarios A and D as inconsistent. And we proceeded and further explored the scenario B and the scenario C. You can see here scenario B, spatially concentrated pattern and high penetration of technology. That means offshore technology-intensive aquaculture, which promote this offshore prototype of aquaculture development. And you have in this axis, the spatial dimension, large aquaculture businesses, 4 to 5 units in the Greek seas, and full integration of mature technology at all stages of the production, full automated. And of course, we have here major investors. What are the benefits? Sector's competitiveness, strengthening the country's leading position, market expansion, creating added value and competitive in cost terms. This scenario set decentralized spatial pattern and medium-low technology penetration, labor-intensive, locally based aquaculture. This has limited potential for innovation, spatially scattered aquaculture model, and in this axis, spatial dimension, we have larger number of aquaculture units all over Greece, low or moderate technology penetration, large number through smaller and weaker businesses placed. This is less powerful, but it can play a complementary role for better utilization of marine resources. It is in harmony with the current pattern of development of the aquaculture sector, and it is competitive in terms of quality of the product. The evaluation takes place here. The criteria are derived from the key drivers from the spatial pattern, and the score is displayed. The rating is qualitative and has been derived from the knowledge of the authors and studies from the international literature. Scenario B is very efficient, very effective in the first criteria, and it has, while the scenario C shows poor performance in this criteria. And it has a good performance in local development, social and economic cohesion. You can see this diagram, and here are the results. Scenario B, better performance in the competitiveness and extroversion of the aquaculture sector, while scenario C has better performance in pursuing local and regional development objectives. Both scenarios contribute to aquaculture development, each one in its own way. They have complementarity, and our proposal is the exploitation of the combination of the two scenarios, B and C. So, in conclusion, or at the end, we can see here the triangle, debating the future. We have the thinking and the shaping the future. So, what is the future of aquaculture in Greece? A political dilemma to be solved. A high-tech and competitive offshore model competing on lower cost, or a model or more dispersed identity-based model competing on traditional and qualitative products? A policy questions that needs to be negotiated in the context of revising the spatial framework for spatial planning and sustainable development in aquaculture. Based on the results, the foresight was very useful. It gave the ability to build plausible images of the aquaculture future in Greek seas. Thank you.
Any question? Okay.
Oh, I really liked your presentation. I have a question. Can both of these scenarios coexist in parallel?
Yes, they can coexist because one of the scenario with the concentrated pattern is this scenario that already exists in Greece. But we can create other sites for offshore development of aquaculture activities and creating synergies with other sectors like renewable energy sources. And there is an interesting. The technology is developing, and we can think about it for the near future. Because this spatial framework that is under revision, the last one was issued in 2011. Now we are so many years after. So if this will be revised and the results will be published in 2 or 3 years, let's say from now, then it will have an horizon for 10 years after. So we have to explore all these new possibilities. Thank you.
Thank you, Mr. Argyro. Thank you also, all of you. There is? Okay, sorry.
Thank you. Mr. Argyro, thank you very much for this very interesting presentation about the future of aquaculture in Greece and the scenarios that you presented. And I wonder how these scenarios can be a part of the policy that the government is going to adapt in the future.
This is the foresight exercise. We have analyzed the first phase, the debating the future, but there are also some other phases thinking about the future and shaping the future. We have done a preliminary analysis and we have a lot of thoughts under the table, but this is going to be explored through the revision of the spatial framework for aquaculture. So when we say thinking of the future, there was the triangle. The one corner is the debating, the other is the thinking. You have to do insights into the sector, to discuss with people, to see what exactly they want, what are the conflicts with the other sectors, are there environmental impacts, can we minimize them? And after all this thinking and exchange views or stakeholder engagement, you can shape the future through the policies. When you have the images, you can shape the future. You can shape it from now. You can shape through the policies to prepare them in advance.
I wonder, in case that it is selected the scenario that we're going to follow, we're going to have big companies that they're going to actually, and not the scattered scenarios. Is there enough, is there, regarding the marine spatial planning, areas that can serve these big companies of aquaculture?
Could you please repeat, are there
The scenario, because you presented some scenarios. One scenario is to have plenty small aquaculture companies.
There are a lot of them all over Greece.
Yes, or to have more concentrated, bigger companies. So these larger companies.
There are large companies now. We have a dialogue. Never mind. There are large companies, but they don't have the ability to build larger farms because we don't have the plan. We don't have the spatial plan. We have to create in order to build, in order for investors to come to Greece, or investors or Greek investors.
And from your experience, do you think there are areas in Greece, in the marine environment of Greece, that they can serve these big investments?
Yes, we did a preliminary approach. We have found such an areas in some parts, in some parts of Greece. But okay, it was not the purpose of this of the present work. We have placed them on the map also.
Okay, thank you.
Okay, thank you again. Thank you to you all. Finished.
Thank you very much for your questions. Yes, because I studied for the sector and for this work a lot of time. I invested Yes, I put a lot of effort, so thank you.
Now, I think now it's okay. Ακούγεται. Να το ρυθμίσουμε τώρα, να είμαστε... Ναι. Ακούγεται τώρα. Όχι. Μόνο επιστροφή. Δεν κοιτάω. Όχι, να σηκώσουμε την κάμερα. Εντάξει. Δοκιμή. Can you hear me now? Okay. I think it's okay from this distance. It's okay? All right, thank you. We are ready to start our opening ceremony. Just for the flow note. My name is Michael Hadjievstathiou. I'm the chairman of the Congress, and I want to welcome everyone here in this room. But first, just to know, we'll have a short ceremony for the beginning, then for introductory, then we have the awarding ceremony, and after that, we have a meeting outside with folklore dances for everyone to know each other. So, dear colleagues and friends, I'm very pleased to welcome all of you tonight in Mytilene at the premises of the University of Aegean and the hospitable island of Lesbos to attend our fifth Congress. The main theme of HydroMediT 2024, we decided to focus on the integration of research and scientific collaboration on our effort to foster the sustainability of the blue economy and to highlight the need for policymakers, industry players, and scientists to work together and deal with the different challenges of blue economy. But please, let me be a little bit more personal. This year concluded almost 30 years from the first time I visited Lesbos, 29 to be precise. I was then a young researcher, and this was my first work on the field. I took part on the study of the National Center for Marine Research for the protection of Kalonic Gulf. 10 years after, following another study break in Portsmouth, I came back to the island, this time to spend here 2 full years doing research for my PhD on the sustainable development of island regions. So, I'm really moved to see today in the audience a lot of friends and colleagues from institute and places where I spent many years of my life trying always to promote our collaboration. I have faith that our HydroMediT 2024 Congress will bring together in its sessions different experience from our region and its adjacent seas, while offering many networking opportunities. Finally, being the chairman of this Congress, it is my honor and pleasure to welcome you to HydroMediT 24 on behalf of our 4 organizers. The Department of Ichthyology and Aquatic Environment, University of Thessaly, and I welcome Professor Exadaktylos here as its representative. The Department of Fisheries and Aquaculture from University of Patras. Professor Moutopoulos will join us in a few minutes. And the Department of Marine Sciences from the University of Aegean, that is our host institute, and Professor Koutsoumbas. And the Panhellenic Society of Technologists Ichthyologists, from where I also come, and its president, Dr. Nikolaos Vlachos, who is here with us in the audience. And with finishing this short introductory speech, I want to, we have greetings from the Minister of Rural Development and Food, Mr. Lefteris Savgenakis. It's a short greeting that I'll now read to you. Dear ladies and gentlemen, thank you very much for the invitation to HydroMediT 24 conference. I'm sorry that I cannot be among you. The HydroMediT conference is now a forum for the gathering of the research world and beyond. From 2018 to date, it has managed within just a few events to be a beacon for discussion and for further development of the pillar of blue economy. With internationally renowned scientists and the participation of national and European institutions, it is developing a dynamic debate between scientific documentation and practical application, while covering a range of topics around fisheries and aquaculture issues. The policies and programs of the Ministry of Rural Development and Food under the Common Fisheries Policy emphasizes and strengthens this important part of the primary sector. We are processing now with the establishment of interbranch organizations in the sector of fisheries and aquaculture, which will strengthen the competitiveness of the fisheries products. In addition, the 2021 to 2027 operational program for fisheries and aquaculture, with a total budget of 520 million euros, covers interventions that contribute to facilitating a sustainable blue economy in coastal, island, and inland areas, and promoting the sustainable development of fishing communities and aquaculture communities, achieving economic and social benefits and broader biodiversity and green transition objectives. I wish you every success in the work of your Congress, and I look forward with great interest to be informed on the conclusion of your work. Best regards, Lefteris Savgenakis. Now, I thank you again for your participation, and I pass the floor to the Vice President of the Congress, Professor Koutsoumbas.
Dear colleagues, dear friends, Rector of the University of the Aegean, Dean of the School of Environment, dear colleagues, dear participants of this event, being the host organization as University of the Aegean as Department of Marine Science, I'm really happy to welcome you all here in this fifth HydroMediT 2024 Congress, a Congress which, as already has been mentioned by the chairman of the president of the Congress, is dealing with different thematic areas, scientific areas related to oceans and in general inland water. I think everybody knows the significance of our oceans and our seas, especially in the last period. I would say that international and global events have been hosted in Greece. Very recently, we had the 9th Our Ocean Conference held in Athens, where I think people from 110 countries all over the world have discussed for 3 days for issues related to oceans and the seas. So this conference is really coming in this, let's say, direction to host issues related to the oceans, the seas, and the inland waters in general, in different aspects, in fisheries, in aquaculture, in marine biodiversity, in ecology, in environmental education, in inland waters, and apologies, I forget something. It is also really an honor for us host this event since it is a long journey which has started many years ago. And I think the important issue is the fact that with this event, we, let's say, I think we managed to show the character not only of the department, but the whole University of the Aegean, is to try to have, let's say, in collaboration with other organizations, it is always, HydroMediT is a conference which is, you know, every time it's coming in the floor, it has more and more participants and organizers. So you can see from what the chairman, the head of the Congress, Dr. Hadjirsafiou has also mentioned earlier, University of the Aegean, University of Thessaly, University of Patras, Ichthyologists, Greek, and also we have supporting other organizations, and this shows that we're trying to be in the heart of events related and bring it all together in this, I hope, interesting conference for all of you. So I welcome you on behalf of the Department of Marine Science. And before we call in the floor people from the University of the Aegean, you allow us to give just very quickly the floor to the other organizing organizations. I see here in the floor Professor Thanasis Exadaktylos. So please, Thanasis, before we go on and call other people from the University of the Aegean, please.
Good afternoon, everybody. I'm Thanasis Exadaktylos, representative from the University of Thessaly and the Board of Directors of the University. I'm really delightfully glad to be here in Lesbos yet again after 22 years. Brings back great memories from my academic career at its beginning, and blend with friends and colleagues that I met yet again. Back to the future now, the University of the Aegean has grown bigger, has grown stronger, has grown prettier, full of facilities to host such international events as HydroMediT. Therefore, I thank the rector for his hospitality, the head of the department, and the head of the, of course, chairman of the Congress for organizing this event. I wish all the best to all delegates, invited keynote speakers, presenters, and especially students that are present here, and Artion for the organization. Make the best of it, enjoy the event with smiling faces, seize every moment of the days here in Lesbos. Our fifth HydroMediT is already a success, as I see it by this morning. Thanks to you all being present here. The floor is yours. I welcome you.
I will call the floor the rector of the University of the Aegean, Professor Dimitris Papageorgiou.
Well, thank you very much, President. Thank you, all of you, for being here. I would like to welcome you in the 5th International Congress or Conference on Applied Ichthyology, Oceanography, and Aquatic Environment, HydroMediT 2024. And I would like to underline that HydroMediT has been established as one of the major international congresses or conferences on novel scientific and technological trends and aspects on applied ichthyology, oceanography, and aquatic environment, covering the Mediterranean and the adjacent seas, with an increasing representation of the academic community. We're very glad that this conference and congress will take place in Mytilene, and we wish that you will have a chance to meet also local hospitality and perfect points, perfect stance from the local environment. Speaking about the guests we have from other countries and from other areas of Greece also. This includes you, Thanasi. So I wish the best and have a fruitful conference. Thank you very much.
And I will call to the floor Professor Panagiotis Mitrakopoulos, who is the Dean of the School of Environment, which has 3 departments, with the Department of Environment, Marine Science, the Department of Diets. Please.
Ladies and gentlemen, esteemed colleagues, member of the organizing committee, and honored guests, it is my great pleasure to welcome you to the 5th International Conference on Applied Ichthyology, Oceanography, and Aquatic Environment under the important theme on integrated research and scientific collaboration to foster sustainability of the blue economy. Our shared mission is crucial to balance economic growth with the conservation of aquatic ecosystems. Over the coming days, let's share our research, forge new collaboration, and drive forward innovative solutions that will ensure a sustainable future for our oceans and waterways. Thank you for your commitment to this vital cause, and let's work together to make a positive impact. Many thanks and welcome to the Congress.
Now we call on the floor Professor Moutopoulos from the University of Patras at Metzologic Campus as one of the organizers to give us also a short greeting.
Good evening to everybody. Thank you very much for your attendance. We are very happy meeting you here after a period of pandemic issues, after 6 years of meeting you. I hope that now we are also start to begin our mutual collaboration, and this conference will also provide as always, with AIM and all the support for the sector of fisheries and aquaculture. Thank you very much for your support.
Now we proceed with the awarding ceremony. We call on the floor Professor Drossos Koutsoupas to give the first award to Emeritus Professor Michalis Karidis.
Dear colleagues, dear friends, allow me to read and say some words for our first awarded representative of the scientific community, which is Professor Emeritus Michalis Karidis. So, Michalis Karidis has studied natural sciences in the University of Athens, where he took his bachelor in 1973. Then he has been a PhD student in the hydrobiology laboratory of the Nuclear Research Center Democritus, specializing in the use of tracing techniques, focusing on the problem of eutrophication. He continued in the Department of Marine Science of the University College of North Wales, UK, where he was awarded his PhD degree in 1978. He has worked as a research fellow in the National Research Center Democritus during the period 1980 to 1992. His academic career was continued in the Hydrobiological Station in Rhodes as a director for 3 years, 1983-1986, being one of the founding members of the University of the Aegean. He was appointed professor in the Department of Environmental Studies in 1998. Michalis Karidis was commissioned by the Senate of the University to organize the Department of Marine Sciences. He has been the head of the department from its beginning back in 1999 for a decade. He has retired in 2017, being since then Professor Emeritus. His research interests, apart from marine eutrophication, include phytoplankton ecology and physiology, aquatic toxicology, coastal management, and over the last 10 years, Professor Karidis is working on issues of marine spatial planning. In addition to his work published in international journals, Michalis Karidis has written books relevant to the marine environment. He is included consistently over the last decade in the upper 2% of the most known researchers in the field of his research. He has also been a member of the National Council of Research and Technology, and within the framework of Erasmus program, he has lectured in European universities in UK, Germany, Poland, and Finland. He spends his leisure sailing, painting, and hiking. And I would add here also keep on going writing scientific papers. Out of the text, you allow me, since we have been together with Michalis over the last 25 years from the beginning of this department, to say some things coming from my heart. The most important issue with Michalis is that he has been inspired as an oceanography to create a department in Greece, which is a unique department offering undergraduate studies in oceanography and marine sciences. And I think he has a long track in oceanography in general and in marine sciences. And the most characterizing issue for him is that he tried, and this is an inspiration which us, his colleagues, we think that we have keep on going in the
initiative he has started from the beginning of this department, Department of Marine Science, to have this multi-approach in issues and trying to bring together all aspects of oceanography and marine science, because this comprehensive approach is the right approach. So in giving this award is a really pleasure and an honor for all of us. And I express not only the people and his colleagues from the Department of Marine Science, but I do think that I also express the whole university. You heard earlier that he was of the early beginners of this nice trip in the University of the Aegean, and also of the marine science community of all Greece. So I will call him in the floor to say some words. Michalis, please, before we offer the award to him. Michalis, the floor is yours.
Good evening, ladies and gentlemen. I would like to thank the organizing committee for the prestigious award to me for academic performance. And also I would like to thank Professor Koutsoubas for his good words he's just said for me. I'll get the opportunity to read you my thoughts. It will be a short speech regarding the development of marine sciences in Greece. I must say that I've started as a research student in '73 in the hydrobiology laboratory in Demokritos, and up to date, it's 51 years of work, which is slightly more than half a century. When I've started my graduate studies as a PhD student in the Nuclear Research Center Demokritos, beginning in the '70s, there was only a handful of marine scientists in Greece qualified that had studied abroad in well-established marine science institutions. Since the '80s, though, the growth of marine research in my country developed exponentially. MSc courses were established in many Greek universities. The National Center for Marine Research was reorganized and further developed, and at the same time, the Marine Research Center in Crete was founded. Meanwhile, Greece managed to build 2 research vessels. Many university departments appointed academic staff specializing in various scientific disciplines regarding oceanography, and their university syllabus included subjects covering many different aspects of the marine environment. At the same time, many more scientific disciplines were added to the so-called classic sectors of marine science. I will just mention you a few examples: satellite imagery and GIS applications, environmental management, marine biotechnology, marine ecosystem services, advances in ocean drilling, advances in aquaculture research and practices, enhancement of the legal weaponry regarding actions in the marine environment and environmental protection. And over the last 10 years, marine spatial planning. Concurrently, the scientific manpower providing services in both the public and private sectors has increased in a number of employees, but also in the academic quality. It is my greatest fulfillment to see that the generations of marine researchers that followed me have better qualifications and are more productive compared to the standards of my time. Thank you again for the award and wish you every success with your conference.
The page down. Oh, yeah. Okay. Yeah. Right. Now, we continue with the second award to a professional, and this will be Mrs. Ioanna Argyroux, a biologist, ichthyologist. Mrs. Argyroux has studies in biology from Aristotle University of Thessaloniki, a master's degree on environment and development from the National Technical University of Athens, is a consultant of operational development and a founder and CEO of the company named NAVS. She is a consultant and technical advisor in fisheries, aquaculture, environment, renewable and resource, and she has over 30 years of experience in conducting and implementing studies, management, and evaluation of investments. She has many scientific publications and presentations in national and international conferences. It is a member of the Independent Expert Database for European Research and Innovation. She is also a co-founder of the Hellenic Technology Platform for Aquaculture and participant and administrator of the patent entitled Ichthyology, aiming at the protection and growth of the fish population. She has been the founder and CEO of NAVS company for the last 26 years, a company that is specialized in studies and consulting services in the field of sustainable fisheries and aquaculture, environmental and social monitoring, climate change, environmental policy, and strategic environmental impact assessment. Through her company, she is engaged in marine spatial planning and blue growth, in circular economy issues, in green development, while it has also been providing relevant services in alternative tourism, marketing, standardization, and processing of agricultural products. So through her whole career, all these years, she was one of the significant factors for the success of Greek marine aquaculture. So please, Mrs. Argyroux, come to the floor and join us, give us a short video.
Good afternoon. I would like to thank the organizing committee of the Congress, its chairman, and the organizers of the Congress. It is my honor and pleasure to be here and be awarded. I also would like to thank the colleagues who trust me and my company NAVS. I have been working in fisheries and aquaculture sector for 30 years. Fisheries and aquaculture is a very important sector for coastal communities. Research and technology, innovation, and relevant policies could support the competitiveness, the sustainability, and the extroversion of the sector for the improvement of the environmental aspects and the acceptance of the sector from the public. Θα ήθελα να συνεχίσω στα ελληνικά. Μεγάλη ήταν και είναι η αγάπη μου για τη θάλασσα, για την Ελλάδα και φυσικά για τον κλάδο της αλιείας και της υδατοκαλλιέργειας. Εύχομαι λοιπόν με το βλέμμα πάντα στο μέλλον να κοιτάμε πάντα πιο μακριά, στο διαυγή ουρανό, σε όλους τους ορίζοντες, σε όλες τις κατευθύνσεις, για να έλθει η άνοιξη στις σκέψεις, τις σχέσεις, τις επιλογές, τις επιδιώξεις, για την είσοδο στην άνοιξη, την επανείσοδο, την επανεκκίνηση του δημιουργικού κύκλου του κλάδου. Το κάθε καλό ας γίνεται καλύτερο. Let everything good become better. Thank you very much for the award.
Our last award person is coming from the small-scale fisheries. Ο τελευταίος βραβευτής στη σημερινή μας τελετή βραβεύσεων ανθρώπων από το χώρο της θάλασσας είναι ο ψαράς μας, ο κύριος Παναγιώτης Νανίδης. I will make maybe it's not necessary. You can read the English, so I can speak in Greek. Okay. Since I think you can read about his professional career. Ο κύριος Νανίδης λοιπόν ζει στο ψαροχώρι τον κόλπο της Καλονής, τον αγαπημένο του κόλπο της Καλονής. Έχει υπάρξει για πολλά χρόνια ο πρόεδρος του τοπικού συλλόγου αλλιέων. Την περίοδο 2013-19 ήταν μάλιστα ο πρόεδρος των μικρών επαγγελματιών αλλιέων ολόκληρου του νησιού της Λέσβου και από το 2019 μέχρι σήμερα είναι ο πρόεδρος του Συλλόγου Μικρών Παράκτων Αλλιέων Μάκαρ, που είναι στον κόλπο της Καλονής. Αυτό που είναι σημαντικό για τον κύριο Νανίδη, στο πρόσωπο του οποίου βραβεύουμε τους μικρούς επαγγελματίες αλλιείς, είναι το γεγονός ότι πάντοτε σε όλη του την καριέρα, τόσο σε ό,τι αφορά την δράση του στο πεδίο, δηλαδή στη φύση, στον κόλπο της Καλονής, τον αγαπημένο του, όπου Οι βιολογικοί πόροι, οι θαλάσσιοι, τους οποίους συλλέγει είναι ψάρια κυρίως και ασπόνδυλα, πάντοτε στην προσέγγιση του, όπως και γενικότερα των ανθρώπων που είναι στο Σύλλογο Μάκαρ, ήταν να υπάρχει μια βιώσιμη διαχείριση των αποθεμάτων των θαλάσσιων βιολογικών πόρων. Και όχι μόνο στα λόγια, αλλά και στην πράξη. Πάντοτε σε όλα τα φόρα όπου έχει κληθεί να μιλήσει, πάντοτε και σε επίπεδο συνεργασίας και με τη Διεύθυνση Αλίας, αλλά και με την επιστημονική κοινότητα στο νησί, το Τμήμα Οκεανογραφίας και Θαλασσίων Βιοεπιστημών, επιστημών της θάλασσας παλιότερα, πάντοτε στο πρόσωπο του αγαπητού μας κύριου Νανίδη βρίσκαμε έναν άνθρωπο με τον οποίο μπορούσαμε να συζητήσουμε και να αλληλοκατανοηθούμε σε θέματα που έχουν να κάνουν με την ορθολογική διαχείριση, τη βιώσιμη διαχείριση των πόρων. Γιατί, ως γνωστόν, αυτό είναι που είναι το σημαντικό. Αυτούς τους βιολογικούς πόρους και αυτή την φυσικότητα των οικοστημάτων πρέπει να την παραδώσουμε και στους απόγονους μας. Στο όνομα λοιπόν, στο πρόσωπο του κ. Νανίδη λοιπόν, βραβεύουμε όλους τους ανθρώπους που η σκέψη τους διέπεται από αυτά που μόλις προείπα, την ορθολογική και βιώσιμη διαχείριση των θαλάσσιων βιολογικών πόρων. Να καλέσω τον αγαπημένο μας κ. Νανίδη να ανέβει στο βήμα και να μας πει και αυτός δύο λόγια.
Είναι μεγάλη μου τιμή να βρίσκομαι σήμερα εδώ πέρα σε αυτή την ευχάριστη αίθουσα. Θα ήθελα να χαιρετήσω όλο εδώ πέρα το πάνελ, από τις κυρίες και τους κυρίους. Είμαι ο Νανίδης ο Παναγιώτης, 68 χρονών. Το 1963 έκανα τα πρώτα βήματα αεραστεχνικά, όταν έκλειναν έτσι τα σχολεία. Όχι τα σχολεία, το δημοτικό σχολείο, το οποίο πήγαινα. Και από το 1967 μπήκα στον επαγγελματικό τομέα σε ό,τι αφορά την αλεία. Είχα την ατυχία να μην μάθω γράμματα, διότι ήταν άλλες υποχρεώσεις, αλλά είχα και τη μεγάλη τύχη σε όλη μου αυτή τη διαδρομή να παραμείνω στο νησί μου, σε ένα νησί το οποίο έχει δύο καρδιές, τον κόλπο Καλονής και τον κόλπο Γέρας, και να απολαύσω γενικότερα όλα αυτά τα φυσικά φαινόμενα, έτσι που κατά καιρούς άλλαζαν. Θα ήθελα επίσης να ευχαριστήσω γενικότερα το Πανεπιστήμιο Αιγαίο και ιδιαίτερα τους καθηγητές, τον κύριο Κουτσούμπα Δρόσο και τον κύριο Γιάννη Ζάκα, ναι, για τη διάκριση και την πρόταση την οποία μου έκαναν. Θα ήθελα να ευχαριστήσω όλους τους συναδέλφους μου τη δεκαετία του 90 και ιδιαίτερα από το 2013 μέχρι το 2019 και από το 2019 μέχρι και σήμερα, που με τη ψήφο τους με είχαν και με έχουν επικεφαλής για να δίνουμε έναν πάρα πολύ μεγάλο αγώνα γενικότερα για το αύριο των ελληνικών θαλασσών. Οι ελληνικές θάλασσες έχουν αύριο, έχουν μέλλον, έτσι γενικότερα και για όλα τα νέα τα παιδιά. Βέβαια, οι προτάσεις μας, σε ό,τι αφορά με τις πάρα πολύ μεγάλες αλλαγές που έχουν γίνει και με τις ανακατατάξεις γενικότερα στην αλληλεία, στο περιβάλλον, κλιματικές αλλαγές, τι θέλουμε, τι θέλουν οι ελληνικές θάλασσες. Είμαστε στην πρώτη γραμμή, πάντα και προφορικά και γραπτά, γενικότερα και στα Υπουργεία και στα θεσμικά όργανα τα οποία εκπροσωπούν το νησί. Τι ζητάει, βέβαια, μέχρι και σήμερα, ίσως να κοφεύουν και για άλλους διαφόρους λόγους. Εμείς αυτό που ζητάμε και στοχεύουμε είναι να υπάρχουν οι ισορροπίες γενικότερα ώστε να μπορούν να επιβιώσουν και οι άνθρωποι και τα θηλαστικά και τα πουλιά που τα έχουμε παρέα σε καθημερινή βάση. Θα μπορούσα να μιλάω πάρα πολλές ώρες για τη θάλασσα με επιχειρήματα μη ανατρέψιμα. Βέβαια, δεν είναι επί του παρόντος. Εγώ αυτό που ήθελα να πω για ακόμα μια φορά, να ευχαριστήσω γενικότερα το Πανεπιστήμιο Αιγαίου και τους καθηγητές και εσάς που με ακούσατε έστω και για λίγο. Σας ευχαριστώ πάρα πολύ.
After that, we can proceed to go outside to the foyer. We have a wine and some delicacies from the region of North Mediterranean. Thank you.